Hello, everyone. Very good evening to you all. Can you all hear me? Can I have some quick responses in the chat? Good evening, everybody. So, this is Tithi here, and today I'm going to discuss the questions. Yesterday the question paper was released from CSIR and the provisional answer key was released. So we have tried to solve it as soon as possible, and here I am with the questions from Unit 3, Unit 8, and Unit 11. Okay, so I'll be mixing the questions from morning shift as well as evening shift. So one thing that I noticed in the paper pattern, okay, so most of you have, yeah, so beautiful colors. And Venkatesh, good evening, welcome to the session. So which shift did you people attend to? Nagashri, good evening. Which shift did you people attend to? I hope all of you have checked the provisional answer key and you have checked it with your score also. Yes, but so far the questions that I have solved, so there is one question that I have found where you can do a challenge. If you have attempted that question, you can do a challenge for that because the options were given wrong. Yes, so I will tell you during my session. Yes, so which shift did you people attend? Venkatesh, Nagashri, beautiful colors. Can you let me know? Yeah, others also. Which shift? So we are simultaneously solving the questions from morning shift as well as evening shift. So today we have a 3-hour session. So after me, Urmimala ma'am will come for discussing the questions that came from Unit 1 and Unit 5. And then after that, Divya ma'am will come for discussing questions from cell biology, cell communication, plant physiology, and as well as the Unit 12. Yeah, good evening, Kumar. Good to see you here. Yes, so yeah, so let's get started. So nobody told me which shift. Yeah, beautiful colors, evening shift, right? Okay. So what I was telling you that during the this session also I will tell you what I noticed is that this time they have kind of followed a pattern. Like previous sessions, what I have seen that suppose in a unit if there are 5 topics, so 3 topics are taken. Questions from 3 topics are given in one session. Questions from other 3 topics are given in the other session. Okay, but this time what I saw is they have maintained a similarity. Okay, so if a single question has so same from same topic, 2 different but related question has come in morning shift and evening shift. So this is a kind of thing that I saw here this time, at least for unit 3, 8, and 11 for sure. There are many examples of those kind of questions, and we will see it in during the discussion. Okay, Kumar, all right. So yes, so let's get started. So first one. So this was question from morning shift. So this is talking about mismatch repair system. So in mismatch repair system, you know that what happens that like mismatch repair, basically it happens at the time of DNA replication. And when it is happening, so there will be a parent strand. So this is the parent strand and this is the new daughter strand. Okay. So I'll just change the color. So this is the new daughter strand. Okay. Now, if there is a mismatch, so how will the MMR system identify that in which strand it has to do the correction? So that is identified by the methylation. Okay. So the parent strands will have multiple methylations. Okay. Whereas the newly synthesized daughter strand will have no methylation. Okay. So that way MMR will identify that this is the daughter strand, and whatever changes that it has to do, it will do in the daughter strand. Okay, using the different enzymes which are there for the MMR system. Okay, now the question says that you have a type 1 mutant where it is which has hypermethylation, and because of this the GATC sequence is methylated in the daughter strand as soon as it is synthesized. Okay, and in the second type, GATC sequence in the daughter strand is never methylated. Okay, so what will happen that the strand where it is never methylated means this will be like the normal situation. So MMR system will be able to detect the daughter strand, and if there is any mismatch, it will be able to correct it. But what about the hypermethylation mutant? So in type A, So in type A, both the strands will be methylated. And as both the strands are methylated, MMR system will not be able to detect which is daughter and which is parent. Okay. So it will, so in this case, in this scenario, in type 1, it will accumulate more spontaneous mutations. Okay. So mutations will be more in type A because MMR will not be able to differentiate between parental and the daughter strand. So with this, our answer is option 1. Okay, next we have another question here. So for this next question, so this is a comparison that we have to do between the prokaryotic genes of replication and eukaryotic genes of replication. So first of all, DNA-B. So DNA-B, it unwinds DNA. It acts as DNA helicase at the replication fork. So the work will be similar to MCM complex, which does the same work in the eukaryotic replication. Then we have DNA-C, which will be loading DNA-B on DNA, right? On the DNA. So DNA-C's work, similar work is done by CD6 in case of eukaryote. So B will be 2. Beta clamp. So beta clamp increases the processivity of the prokaryotic DNA polymerase. So when there is beta clamp is there, so the work of beta clamp, the similar work is done by pCNA in the eukaryotic system. And when we have DNA-G, so DNA-G helps in the formation of the primase for synthesizing the primer, okay, to start the DNA replication. So that work in the eukaryote is done by using DNA polymerase alpha with a primase subunit. Okay, so D1. So what we have here, matching A4, B2. Okay, so A4, B2, C3 and D1. Okay, so we have our answer, option 3. So guys, I'm going to go a little faster in explaining you all the questions. So in case if you have any doubt, so our technical team will be posting a form a form link. Okay, so once you fill up the form and mention the question ID, so I will reply you with the detailed explanation of that particular question. Okay, so a form link will be given in the comment box. So in case any question, okay, during the session, my session, or with the followed faculties, any other session, if you have any question that you want a more detailed explanation, so just note the question ID, fill up the form and send it to us. We will reply to you as soon as possible. Next question. Now this is about the this is again from unit 3 and from morning shift I have got. So modifications in the native system that does not lead to translation inhibition. Okay, so this is actually related to, as you see the statements, we are talking about Shine-Dalgarno sequence. So this is related to translation. Okay, so the statements are there which are from prokaryotic as well as eukaryotic translation initiation. Okay, so option 1 says that nucleotide addition resulting in incorporation of stem and loop structure in the mRNA upstream of Shine-Dalgarno sequence. Now, what is the function of Shine-Dalgarno sequence? Shine-Dalgarno sequence, so if we have our mRNA here, so Shine-Dalgarno sequence, which will be present around 8 base pairs, okay, on the 5' side of the initiation codon. Okay, so this will pair with the 16S rRNA. Okay, and pairing of Shine-Dalgarno sequence with the 16S rRNA will position the AUG codon correctly at the P site of the small ribosomal subunit. Right? Now, if there is any nucleotide addition which is there in the 5 upstream means on the 5' side, right, of Shine-Dalgarno sequence. So if there is any stem and loop structure here, will it interfere anywhere in the base pairing of the Shine-Dalgarno sequence with 16S rRNA? Well, no, it will not. Okay, so this will not cause any problem. Shine-Dalgarno sequence can still bind with 16S rRNA. Okay, so most probably our answer is option 1 only, but let's check the other statements. So nucleotide addition resulting in Shine-Dalgarno sequence being a part of stem and loop structure. So Shine-Dalgarno sequence itself If it becomes a part of your stem and loop structure, so definitely it will not base pair. Okay, so this will inhibit translation. Expression of EIF2 mutant that mimics its phosphorylated state, if that happens, again EIF2, when it is phosphorylated, it will not it will become it will become inactive. Okay, so if it is inactive, it will not work and translation will not happen. And option 4, which says that mutation that lead to decrease in the processivity of capping enzyme. Okay, so we know that in case of eukaryotes, 5 prime cap is very important, because that is how the translation initiation factors will bind with the 5 prime cap and bring it to the small ribosomal subunit. So if the 5 prime cap is not there, then mRNA will not be brought. Okay, so translation inhibition will happen. Okay, so option 1 is our answer. Okay, now you see this is the question from evening shift. Okay, and this is also talking about translation, which is basically talking about the primary function of mediator complex in eukaryote. Okay, during gene expression. So in gene expression, we know that in case of eukaryote, the major problem is accessing the DNA. Okay, so where does mediator complex come? So this is the diagram, if you remember. So here the transcription start site is here, the promoter region is here, but the enhancer uh, uh, where the activator proteins will bind, so that enhancer will be many base pairs away from the promoter sequence. So there will be architectural regulator proteins which will bend the DNA so that enhancer can come to closer to the promoter region. Now, at this enhancer, the transcription factors, the activator proteins will bind. Now, this activator proteins cannot directly interact with RNA polymerase at the promoter. Instead, what it will do, it will use mediators or co-activators. Okay, using mediators or co-activators, it will interact with RNA polymerase along with other transcription factors, and that will help in the recruitment of RNA polymerase at the promoter region. Okay, so what is our answer? That it will facilitate the interaction between transcription factor and RNA polymerase. That is the job of mediator. Okay, next question, which of the following translation system is inhibited by cyclohexamide? Okay, so now this is a kind of question which was expected. Translation inhibitors or transcription inhibitors, a question from there is very well expected. Now, cyclohexamide, what it does, it will inhibit 60S ribosomal subunit. Okay, the larger subunit, and this 60 larger eukaryotic subunit. Okay, and because of that, what will it do? It will stop the translocation of ribosome. Okay, so what will be the answer? That it will inhibit the 60S ribosome associated eukaryotic cytosolic translation system. Cyclohexamide does not affect the 70S ribosomal system. So mitochondrial and chloroplast translation will happen normally, but it is in the eukaryotic cytosolic translation that will be inhibited. Okay, now next question from Hardy-Weinberg equilibrium. Again, a very well expected question. In a population with ABO blood group system, if the frequency of allele IA is 0.3, frequency of allele IB is 0.2, so and frequency of small I is 0.5. Okay, and the question is, what is the expected percentage of blood group A? Okay, now this you will be able to answer only when you have the idea of Hardy-Weinberg equilibrium. Along with that, you know how this blood grouping system works. Okay, so we know that IA and IB allele are co-dominant with each other. Okay, and they are dominant over the recessive IO allele. Okay, so who all will have blood group A? Yes, who all will have blood group A? So blood group A, so what all genotypes basically will have blood group A? So either a person has a genotype of IA, homozygous, okay, or if a person has the genotype of IA and small I. Correct? So if you are IA, then also you are blood group A. If you are IA and recessive small I allele, then also you are blood group A. Okay. Question is, what is the expected percentage of blood group A? Okay. So when I'm talking about frequency of group A, so we have to calculate both. Okay. Now here there is a or. So what rule of probability we have to apply? The addition rule of probability. Okay. It is mutually exclusive event. So a person who is of blood group A will either have this genotype or this genotype. Okay. So in that case, it will be frequency of IA plus frequency of IA and small I. Okay. Now our frequencies are given here. So IA's frequency is 0.3. So IA's frequency is 0.3, IB's frequency is 0.2, and small i's frequency 0.5. Okay. So frequency of homozygous, how it is given? P square, correct? So frequency of the allele square multiplied by itself. Okay. So it will be, how much it will be? 0.3 square. Fine. Plus this is heterozygote. So heterozygote frequency is given by what? 2pq. Okay. Now here if we say that this is p, this is q, and this is r. So here the formula for IA small I will be 2pr. Okay. Frequency of IA allele multiplied with the frequency of the recessive allele small I multiplied with 2. Okay. So when you substitute here, it will become 2 multiplied by 0.3 multiplied by 0.5. Okay. So when you solve all of this, you will get 0.39. And when you convert it into percentage, you'll get 39%. I hope everyone getting it. Yes. Now, next question. Now, this is from evening shift, and you see a similar question again about Hardy-Weinberg equilibrium. But the question is not so simple that you can just respond, answer it by Hardy-Weinberg equilibrium. You also have to have a basic idea of genetics here. Okay. So it is saying that in a population, frequency of allele A is 0.2, frequency of allele B is 0.1. Consider there are 2 alleles for each of the genes. Okay. Means this is one gene. Let's say this is gene 1 and this is gene 2. Right. So gene 1, if small a is one allele, so we have to suppose what? That it has another allele which is capital A. Similarly, if gene 2, one allele is small b, so we have to suppose that there is another allele which is capital B. Okay. Now, if small a's frequency is given as 0.2, what will be the frequency of capital A? Capital A's frequency will be 1 minus 0.2, right? That will become 0.8. Similarly, if we are talking about frequency of small b allele, now if that is 0.1, what will be the frequency of capital B allele? 1 minus 0.1. So it will become as 0.9. Fine. Now, what would you expect the percentage of population with genotype capital A, small a, capital B, small b? Okay. Now, for capital A, small a, so here what we have to do, frequency of capital A, small a, capital B, small b. Okay. Now, since they are 2 different genes and nothing is mentioned about they being linked or not, so we'll consider them as independent events. Okay. So if they are independent events, right? So how we can break this down? It can be broken into the 2 combined events, right? So 2 independent events. So frequency of the combined event is given by the multiplication of their individual frequencies, right? So what is the frequency of heterozygote? What is the frequency of the B alleles heterozygote, right? Similar thing we used to do in the Mendelian cross also. So now what will come here? So what will be the frequency of this A? So for that, so suppose I have given that this is the PA and this is QA, okay, and this is PB and this is QB. Fine. So what will be the frequency of this heterozygote? It is given by 2PA multiplied by QA, 2PQ, okay, whole multiplied by, again for B gene also it will be 2PQ, so 2PB multiplied by QB. Fine. Now substitute it here. So 2 multiplied by 0.8 multiplied by 0.2, and this multiplied by 2 multiplied by 0.9 multiplied by 0.1. Okay. Now when you solve this, okay, so what will you get is, yeah, please go ahead and solve it. So it will come as 0.32 this side, and this will be multiplied with 0.18. Now answer that you will get will be 0.0576. When you convert it into percentage, that means you have to multiply it with 100, right? So that will come as 5.76%. Now this is the question that I was telling you, that according to answer key it says option 4, which is this, but you see the numbers are same but decimal point is at somewhere else. Okay, and definitely with the allele frequencies that is given and the genotype which is given here, so the frequency of that cannot be this high. All right, now this is the question where you can do your you can do a rechecking. Okay, you can challenge this question. So remember, the question ID is this. If you have attempted this question, and please go ahead and challenge it. Okay, so answer given is option 4, but option 4 is incorrect. In fact all the options are incorrect. So here instead of 57.6 it should have been 5.76%. Right, moving on. Next question from morning shift. I have the mutation in a Drosophila gene. Okay, reduction of eye color. Okay, so what we have here, so if animal are grown in 29 degrees Celsius, okay, but not at 18 degrees Celsius. So there is a reduction in eye color. So you can write it down, 29 degrees Celsius, reduction in size. 18 degrees Celsius, it is normal. So this happens even if the animal bear a single copy of the mutation. Based on the information, the mutation is described as. Okay, so very first thing that you can see is that the mutation is behaving differently at different temperature. This tells us about a temperature sensitive mutation, right? Or even a conditional mutation, right? So definitely it has to be temperature sensitive. Now you can see whether it is dominant or recessive. So very clearly it is mentioned that single copy of the mutation will still cause a reduction in the eye size. Okay, single copy. What kind of allele are we talking about? Definitely a dominant allele, because a single copy of that can still make it inactive. Right? So answer will be option 4. Okay, now this is again a question like temperature sensitive mutation, something related to conditional mutation or temperature sensitive mutation we are seeing very commonly asked nowadays. So definitely this will be a topic that you have to focus for next time. Yes, so in the morning shift, I noticed that from unit 3, lot of part C questions came. from unit 3, lot of operon related questions came, okay, and very interesting questions. Now this also is one of those interesting questions. So here it is saying that they have made two different fusion operons, okay. So what they have used is, one is the lambda phage C1 operon, and the other one is the lac operon. Okay, so what they have done, they have put lambda C1 gene under the control of LAC-P and LAC-O, whereas LAC-Z gene is put under the control of the PRM and OR operator of the lambda phage. Okay, now see, for answering this question, you have to have an understanding of how this lambda C1 gene works and also how LAC operon works. Then only you'll be able to answer it. Okay. Now the question is, both the plasmids are introduced in the E. coli and concentration of the proteins are determined upon the addition of IPTG. Okay. Now what will IPTG do? IPTG is a gratuitous inducer of lac operon. Okay. So IPTG will induce lac operon. So when IPTG is present, so IPTG will go here, bind with the inhibitor of the lac operon, remove it, and the lac operon will be on. Okay, now lac operon is on. Now when this lac promoter, like from the operator region, when the inducer is removed, so here it is not LAC-Z gene that it is controlling. What it is controlling? Lambda C1. So lambda C1 will be expressed. Okay, so lambda C1 repressor, right? So what we see, as you can see here, IPTG. So as the concentration of IPTG increases, lambda C1's concentration will keep on increasing. Okay, so that is the same curve that you see in all the 4 graphs. Okay, the blue line is same. Fine. Now next, what will this C1 repressor do? Now this C1 repressor, so it will interact with the PRM and OR regulatory region. Okay, so what happens is, when C1's concentration increases, it goes and binds to this ORF2. Okay, now binding of C1 with ORF2 increases the accessibility of RNA polymerase at this PRM, and more RNA polymerase will keep on coming, and the gene that this promoter is controlling, that expression will be there. Okay, now what gene is it controlling? LAC-Z. Okay, so LAC-Z will be now expressed. Fine. Now as LAC-Z is expressed, okay, so what will happen? You see this red line is for LAC-Z. So LAC-Z concentration will keep on increasing. Fine. Now C1 repressor also does an inhibition here. Okay, so when C1's concentration is too high, okay, when C1's concentration is too high, then what C1 do? It will go and bind to ORF3 also. Okay, and binding of C1 with ORF3 will inhibit the operon. Okay, so now when it is binding, so I'll just change the color here, blue. So when C1 is present in high concentration, it will go and bind with ORF3, and that in turn will stop LAC-Z's production. Okay, so as you can see here, so LAC-Z concentration will increase, but after certain concentration of C1, it will get to a plateau. Okay, it will not increase anymore. Fine. So graph C is the answer, option 3. Yes, next question again from the transcription regulation, gene regulation kind of questions from the histone modifications that happen, that is a part of eukaryotic gene regulation. So here there are different statements given and you have to find out the correct statements. So acetylation of histone generally associated with transcription repression. That is wrong, because acetylation will activate the DNA gene, right? Because that makes the DNA loosen up around the histone and more accessible. So acetylation will activate. Methylation of histone can either activate or repress. that is absolutely correct. Okay, but it will depend on which specific residue is modified. In certain if a lysine present at a particular position is methylated, then it will cause activation, whereas a lysine present at some other position, if it is if it is methylated, it might result into repression. Okay, so this statement correct. Phosphorylation of histone occurs in response to DNA damage and can influence gene expression. That is absolutely correct. Okay, so C is also correct, and D says that histone modification influence recruitment of DNA polymerase but not transcription factor. Now remember, in case of eukaryote, RNA polymerase cannot bind with the promoter. It cannot recognize also the promoter until and unless transcription factors bind. Okay, so modification, histone modifications will first influence the recruitment of transcription factors only, and then only RNA polymerase will be recruited. Okay, so statements, correct statements B and C. Answer is option 2. Again, another question from tryptophan operon this time. Now here you should have a good understanding of how tryptophan operon works. Okay, now so let me show you the so this is the working of tryptophan operon. You know that the attenuation mechanism basically. So in case of here, so there are different situations given. So originally what happens that if 3-4 base pairs, then the attenuator structure will be formed. If 2 and 3 base pairs, then there is no attenuation. Okay, so here there is attenuation. Here no attenuation. Okay, now let's see all the conditions. Inserting bases between leader peptide gene and sequence 2. So if there is some extra bases added here, okay, so what will be the use of that? So because of that, the ribosome will always take more time to come from 1 to 2. Okay, so 3 will mostly pair with 2. Okay, and if 2 and 3 base pairs, there is no attenuation. So because of that, there will be less attenuation. Okay, so A condition will lead to less attenuation. Next, inserting base pair between 2 and 3. Now, if base pair between 2 and 3 is there, if insertion happens here, okay, in that case, this 2-3 base pairing will not happen. Okay, and if 2-3 base pairing is not there, then 3 will base pair more with 4, and if 3 base pairs more with 4, so that will result into more attenuation. So B will be 2. More attenuation. Sorry, B will be 3, more attenuation. Next, deleting sequence 4. So if you delete only sequence 4, so 3 has no way of binding with 4 and forming the attenuator structure. So there will be no attenuation at all. So C with 4. D, elimination of ribosome binding site for the leader peptide. So if there is no ribosome binding, means translation is not even starting. Now in that case, 3, when it is transcribed, so 3 inherently has a higher affinity for 4. Okay, so if 3 is transcribed and it has the option of either binding with 2 or 4, it will always prefer 4, and in that case there will be complete attenuation. Okay, so our answer is option 4, A2, B3, C4 and D1. Next, again about lac operon. Now it is saying that in lac operon there are 3 genes which are there, Z, Y and A. It is observed that lac Z is translated more frequently than Y and A. Which one of the following statement best describes it? Okay, so what happens is when we have the lac operon, so lac operon will be transcribed. So here lac operon is transcribed as a single polycistronic mRNA, but that polycistronic mRNA, okay, so if this is the polycistronic mRNA, so that will have individual Shine-Dalgarno sequence for Z. Okay, so suppose Z is getting over, then there will be another sequence for Y, and then there will be another sequence for Z, for A. Okay, so because that is how they are translated into individual polypeptides. Fine. So the reason that Z is present more and Y and A are present less, the reason is because of the variation in the Shine-Dalgarno sequence, which is present in the upstream of Z, Y, and A. Okay, and they have different affinities for the ribosome. Okay, so the Shine-Dalgarno sequence between for Z has more affinity for ribosome, whereas for Y and A, the Shine-Dalgarno sequence that is present has lesser affinity. Okay, and how this affinity is determined? So researchers have found out that this affinity depends on the distance of Shine-Dalgarno sequence between distance of Shine-Dalgarno sequence with the initiation codon. Okay, so this is optimum for Z, so that's why the affinity is more for Z. So answer is option 3. Yes, so again, a question which was very well expected, a question from complementation test came in the morning shift. So 6 yeast mutants are given. Okay, so plus means that so they all were they all were histidine minus. All the mutants were histidine minus. So we basically did a complementation test and see. So if plus is there, it indicates diploid combination yielding histidine prototypes. Okay, so you can see here that half of the table you can completely ignore. So you can ignore either this half or that half. So this will be the middle line. Okay, so now the question is, how many genes are represented among the 6 mutants? Okay, how many different histidine biosynthesis genes are represented? Okay, so how do we calculate that? So complementation test give us the idea that whether the mutations are happening in the same gene or different gene. So if the gene so whatever is the number of complementation group, that will tell you the number of genes which are represented by this 6 mutants, right? So very easy it is. So histidine 1 is not complementing 3 and 6, means 1, 3 and 6 they are present in one group. Histidine 2 is not complementing 4 and 5. Okay, means 2, 4 and 5 are present in one group. Okay, so 1, 2, 3, 4, 5 and 6 all mutants we have placed. So how many groups we have got? Only 2. How many genes they are representing? That is 2. So remember guys, if you people are not able to understand these short answers and you need a detailed explanation, please fill up the form and write the question ID, okay, so that it is easier for us to identify the question and we will send you the detailed explanation. Yes, this question from evening shift again. Okay, so this is a pedigree chart that is given. So for based on the information, statements are given and you have to say whether the statements are correct or not. Okay, so all correct statements you have to identify. First, at least one of the children will carry any mutation linked to this condition. Will not carry. Okay, now see, if you see this particular mutation, right? So it says that both the parents have the mutation, although in the next generation, you have few individuals who are not having the mutant phenotype. Okay, now when can this be possible? See, this is possible if the mutation that is causing this particular particular disease is a dominant mutation. Okay, because if it is a dominant mutation, so let's say A is the causative allele, so that means individual can be capital A small a and capital A small a. In that case, small a are hidden here, so small a alleles can come together and it can form the individuals who are not having the causative mutation. Fine. Now, if you say that it has to be the causative allele has to be recessive, then this situation is not possible because the individuals will be homozygous recessive and all their entire progeny will also be homozygous recessive. Okay, so this is only possible when the mutations are dominant. Now, it says that it involves 2 genes, so both the mutations that we are talking about has to be dominant. Fine. Now, so here it is dominant. So when we have found that it is a dominant mutation that is causing, so automatically option B is incorrect because it says that it is recessive. Fine. Okay, so at least one of the children will not carry. Yes, correct. So where we have these individuals who are not affected, they are the one who are not carrying any of this mutation. This is true. See, at least one of the mutant gene is on Y chromosome. Now that is not possible because in the progeny we see females who are affected. Females will never have any Y chromosome. Okay, and that's why Y chromosome not possible. Simultaneous heterozygous mutation in the involved genes can cause the disease. Yes. So if both the mutations are happening and in heterozygous state also if they are happening. Heterozygous state means it is happening in one homologous chromosome, the other member is not having the mutation. Okay. So now that is possible because the mutation is a dominant mutation. Okay. So statements A and D are correct. Answer option 3. Yes, moving on to next question. Now, this is a very interesting question. It says that mutation in it's from the evening shift. So wherever guys you see E written means it's from evening shift. If it is from morning shift, I have mentioned M. So a mutation in a plant gene is female gametophyte lethal. This mutant allele can be normally transmitted through pollen. However, when transmitted through egg, the embryos abort. Okay. So a student has harvested seed from heterozygous mutant plant, grew a total of 100 plants. What is the expected number of homozygous plants in the population? Okay. So what he has done, he has taken a seed from heterozygous plant. Okay. So this heterozygous plant means the genotype is heterozygote. So let's say we are just take an example of gene A. Okay. So the heterozygote, so when they are crossed, so that is how they will produce this 100 plants. Okay. Now for getting this 100 plants, what we have to like the possible genotypes. Okay. So suppose this is male, and this is female. Okay. Now this female will so what happens here is that it says that the female cannot pass the causative allele. Okay. So causative allele is lethal. Okay. So when it is passed from the female, then it becomes lethal, right? So what is what that now? So the allele. So if we say that the small a is the causative allele. Okay. So when it is transmitted through egg, then it aborts the embryo. Fine. So when we are separating, so you can make the Punnett square, right? So the gametes that, so suppose we are writing the male gametes here. So male gamete will be capital A and small a. Okay. Now we will write the female gametes also here. So female gametes, capital A and small a. Okay. So here it is capital A, capital A, small a. Okay. Now this small a is our causative allele. Okay. Which is the lethal allele, right? So if small a is the lethal allele, then small a will come here and here, and in both the situations, because it is passed from the female parent, so in both the situations it will be lethal. Okay. So this entire proportion of progeny will not survive. Okay. So only this much will form the progeny. Okay. Now in this proportion, so in this progeny, what do you see? So 100 plants that you have, that the student has grown, okay, so they are from this progeny only, where they have got the capital A allele from mother and capital A and small a allele from father. Okay. So in this case, what is the expected number of homozygotes? So 50% are expected to be homozygotes, right? So 50% of 100 will be 50. So expected number of homozygotes here becomes 50. Right. Now, next question is also from the evening shift, which is talking about the various mutagens and the type of change that it can create. Okay. So, among all the options that are given, so all the statements are incorrect, except two is the correct answer, where nitrous acid will deaminate adenine, leading to AT to GC transition. So this was given in part B in evening shift, whereas in morning shift, a similar question was given in part C, okay, where more base mutagens were given and their functions were given. Okay, so and what is the type of the chemical mutagen, and the name of the mutagen was given, and you have to match. So base analog out of all 5-bromouracil is the base analog A5. So this is one of the very easy question actually. Sorry, easy question actually. So A is 4, right? So A with 4. So if you match this also correctly, you have got your answer. You don't even have to match everything. A4, there is only one option with A4. So intercalating agent, ethidium bromide, B3. Deaminating agent, nitrous acid, so C1. And alkylating agent, ethyl methane sulfonate, D2. Okay, answer option 1. Very easy question. Fine. Now, next question. So this is a cross which is given, and again there are statements given, and here you have to say whether the statement is true or false. Okay. Now this cross that is given, so this kind of question has come previously also. Okay. Maybe the format in which that was given was little bit different. Okay. Now, if you see here, so let's see the eye color. So we have to first identify what is the inheritance pattern of eye color and body color. Okay. So eye color, if you see, so in case of eye color, what we are seeing that the father has red eye color, and that red eye is there in daughters as well as in sons. Okay. So that definitely means that the gene for eye color is not present on Y chromosome. Fine, because females will never get it. And also the gene for eye color is not present on the X chromosome also. Okay, because sons will never get the allele from father if it is present on the X chromosome. Okay, that means eye color for sure is autosomal. What about body color? Now for body color, the situation is different. So you see the female is yellow body, and we have the F1 male which is also yellow. Okay. The P male is gray body, and we have F1 female which is gray. Okay, so you see there is a crisscross. Fine. That means body color is present on X chromosome. Okay. Now, as body color is present on X chromosome, okay, so if so, let's see the statement. So mutation leading to sepia eye color is located on autosome. That is true, right? Because eye color is present on autosome. Yellow body is a dominant phenotype. Okay. Now let's see whether yellow body will be a dominant phenotype or not. See here, the female mother is yellow. Okay. Father is gray. All right. Now father has only one allele, but F1 female is a heterozygote. Now whatever is the phenotype shown by the heterozygote, that will be the dominant phenotype, right? That goes in Mendelian principle also. So what phenotype the F1 female is showing? Gray. That means gray is dominant here. Okay. So for body color, if we see say that there is the gene G is responsible. Okay. So small g will, so I'll just write it as XG. Okay, because it is present on X chromosome. So X small g will result into yellow body color, whereas X capital G will result into gray, which is dominant. Fine. So yellow body color is dominant phenotype. This is false. Okay. A true, B false. So with this combination also, you have got your answer. A true, B false. There is only one option with that combination. Okay. Now 1/4 of the F2 progeny will be male with yellow color. So you can do a cross here. Okay. So female. So first you have to write down the genotype. So the genotype of this F1 female will be what? So she is getting capital G, X capital G from father, X small g from mother. Okay. And so you can just write down that for body color, because here it is only talking about body color, not the eye color. So you can just skip the eye color. And for male, it is X small g and the other is Y. Okay. So if you make a Punnett square here, you will see that yes, 1/4 of the progeny will be male with yellow body color. So quickly, if you make it here, capital G, small g, small g, small g, capital G, Y, and small x, small g and y. Okay, so this is the part which is yellow, male with yellow, which is 1/4. So again, this is true. And in this dihybrid cross, as F2 progeny do not show 9:3:3:1 typical Mendelian ratio, two genes can be assumed to be linked. Now we have already established that one gene is present on autosome, one is present on X chromosome. So definitely they cannot be linked. Yes. Now next question from morning shift. Okay. Now here we are moving on to unit 11. So here there are two sympatric ladybird beetle species. Okay, sympatric means in the same area only two different species of beetle have evolved. Question is, which one of the following statement does not explain the speciation process in this beetle species? Okay, so you see in option 3 it says that diverging population developed differences in diet. However, it did not lead to reproductive isolation. Now, we know that for speciation to happen, the very first step that is required is reproductive isolation. Somehow the groups have to be reproductively isolated. Then only what all changes they will accumulate will remain separate, and that is how new species will be formed. Okay. So if did not result into reproductive isolation, that will not cause speciation. Okay. So definitely answer is option 3. Next question. Now see, this is so this was a question in the morning shift. Now this is a similar kind of question which is also talking about reproductive isolation and speciation and came in evening shift. So here you see male sing species specific songs. Okay, now this also is a sympatric species of grasshopper. Males will sing species specific song. Okay, so the song represents which mode of reproductive isolation? So we know that this is a mating. This song is for what? So song is a mating call, right? It is a call to attract the opposite sex. So if the male is singing, it is for attracting the females. Now, since the males of the species are singing different songs, so it will be responded by females of their specific species only. Okay, so what mode of reproductive isolation it is? It will be pre-zygotic isolation, like before the zygote is formed. Now that is not written anywhere, but pre-mating is there. Okay, so definitely it is a pre-mating isolation. Okay, next question. The statements describe change in allele frequency over time as a result of genetic drift. Okay, so what all things are true? So genetic drift, definitely fixation of an allele can happen by chance, while other alleles will be lost. Okay, so this is true. Genetic drift can lead to the loss of certain alleles over time, reducing genetic diversity. That is absolutely true. Changes in allele frequency are due to positive selection. Now, positive selection has nothing to do with genetic drift. Genetic drift happens on chance. It will not take into account whether the allele is helpful or harmful, okay, or neutral. Nothing. It will just happen by chance. So C is not true. D, there is a pronounced effect in small population. Now, we know it very well that effect of genetic drift will be higher in small populations and lesser in larger population. Okay. So answer will be A, B, and D. These are the statements which are correct about genetic drift. Yes, so this is a question that came from the optimal foraging theory, okay, which talks about marginal value theorem, okay, which is one of the model on which the optimal foraging theory is based on. So in this, now a similar type of question came in the evening shift also, and I will show you that as well. So as you see here, okay, so this is the graph that is given, okay. So a major prediction of this theorem is how long an animal must stay in a patch to optimize the energy extracted, depending on its travel time to reach the patch, which is depicted in the figure below. Okay, and in here what you have to do, you have to find out what is P and what is Q. Okay, now this marginal value theorem, it actually, yeah, so that what is mentioned here is absolutely correct. That is what marginal value theorem do. Okay, so in here what happens is, see, an animal will start looking for a prey in a particular area. Okay. Now, the more time it spends there, lesser will be the chance for it to get the prey. Okay. So here you see, this is a graph which is made on the uh, on two axes. So where the y-axis is actually the energy gain. Okay. X-axis is for time. Okay. X-axis is time, y-axis is energy gain. Okay. Now what happens is when animal start looking for its food. Okay. So in a particular area, so this curve, okay, so this is the accumulated energy, so it will start with a steep curve. Okay. So as the time keeps on passing, then the chance of it to get the prey will keep on decreasing. Okay. So at one point it has to decide whether it will stay longer in that particular patch or it will move to a different patch. Okay. Now when it is moving to a different patch, so there will be some energy that it will lose. Okay. And also there will be some foraging time that it will lose. Okay. So whether it has to move that time or not, that we have that can be decided by this marginal value theorem. Okay. So what happens is that this net energy gain is actually given by this diagonal line. Okay. So this diagonal line should be formed in such a way that it touches the curve. Okay. So like a tangent to the curve. Okay. Crosses the energy axis and meets the the time axis here. Okay. So in this case, this p value, okay, the point where it touches the curve, that will show the uh, optimum, it will show the optimum patch residence time. Okay. So the time that the animal has spent in the patch. Okay. Whereas q shows the time taken to travel between a different patch. Okay. So I'll show you the graph. Okay. This is the actual graph, right? Now this has happened again, like previously also, that they have taken some standard graph or some standard image and just eliminated some of the labels and asked you to label. Okay. This is what has happened. So you see here p and q. So this was marked as p, this was marked as q. So p is optimal time spent within patch foraging time. Okay. So how much time the animal spends in that particular patch, and q is the optimal patch. Okay. So answer is option 3. Sorry, option sorry, yeah, time taken. So answer is option 4. Now again, as I was telling you, a similar question, okay, from the same topic is asked in the evening shift. Okay. Which is also talking about optimal foraging theory. Now it says that consider a predator which can forage on two preys, prey 1 and prey 2. Now prey 1 is more profitable than prey 2. Now while searching for prey 1, if it encounters prey 2, so it has to make a decision where it should capture prey 2 and or ignore it and continue its search for prey 1. So that can be given by the predictions of optimal foraging theory. The table below gives the various parameters that may be used as per OFT by the predator in making the foraging decision. Energy gain, handling time, search time. Okay. See, when a predator is looking for the prey, so it will, when it gets the prey, it will get energy. Okay. But it will spend some energy to search for the prey and also from capture to feed. That is called as the handling time. Okay. So this two things will lose energy, this thing it will gain energy. Okay. So prey will be preferred only when this net energy, okay, which is energy gained minus handling, minus the cost of handling and research, that much will, so that will be the net energy gain, and whichever prey has the higher energy gain in that way, that will be preferred. Okay. So in which of the following statement predicts correctly when the predator should eat prey to given the conditions? Okay. So when it should eat prey to in this situation, so basically it should eat prey to when it thinks that the search time for prey 1 is going more, okay, or it is becoming more, then only it should eat prey to, right? Okay, so just like if you have to wait more to get food, right? So if you really want to eat, let's say, a whole Indian meal, okay, with sabzi, roti, everything, but if it takes a lot of energy to make it, all right, so if you think that the time and effort is more in cooking that, instead if you can eat a Maggie, which is unhealthy but still it can be done faster, so in the situation when you have lesser time, you will definitely prefer that only, right? So here also the same thing. So if the predator comes across prey too, which will give lesser energy, but if the time taken, the search time for prey one is very high, then it will capture prey two and forget about prey one. Okay, so S1 has to be high. So option 2 is the only option you see which says that S1 is higher than this value. Fine. So that's why answer will be option 2. Yeah. Now this is the question from reproductive success in birds and parental care, and we are very close to coming to the end of this session, and next session will be taken by Urmimala ma'am. So please stay tuned, everybody. Yeah, so this is a question from parental care and reproductive success. And again, I saw that a similar question came in the evening session also. I will show you that as well. Okay. Now it says that, see the option that identifies the combination of all correct statement. Okay. So if females provide more parental care than males, variance in male reproductive success is significantly greater than that of females. Okay, see, if females are providing more parental care, okay, that means what, the females are occupied in taking care of the young ones, so the males can stray, so they can go and do multiple they can have multiple partners, they can mate with multiple partners. So in that case, males reproductive success will be higher than the females. Okay, this is true. Now, similarly, the vice versa situation is true, that if males provide more, only males are providing the parental care. In that case, females can mate with other partners and they can produce more offspring. Okay, so in that case, the variance in female reproductive success will significantly be higher in the males. Okay, so A and B both are correct. Now C says that in case of biparental care, variance in male reproductive process is significantly greater than that of females. No, that does not happen. If it is a biparental care, then it will be stable. Okay, variance gives you the variation. Okay, but there won't be much variation. It will be more stable. Okay, whether it is for male or whether it is for female, in both the cases, if it is a biparental care that is there, so in both male and female, the variation will be very less. Okay, so both C and D are incorrect. So as I was telling you, a similar question came in evening shift. So here it is, male mating systems in bird. Okay, it uses the number of successful matings as a measure of male reproductive fitness and female reproductive fitness. Okay, and it is given by P and Q. So P is the black bar, Q is the gray bar. Which of the following option correctly match P and Q with the correct sex for different mating systems? Okay. Now the similar situation will happen. So if so, that was related to parental care. Now this will be related to the mating system. Okay. So if there is polygyny, polygyny means what? Polygyny. Polygyny means males can mate with multiple, males will have multiple partners. Okay, so if males have multiple partners, then the variation, so one person, so one male can get multiple female partners. Okay, so they will produce more offspring, whereas at the same time there might happen that the male a male has got very few partners. Okay, so in that case, number of offspring produced will be less. So basically, when there is polygyny, when males have multiple partners, so in that case, variation in the reproductive fitness will be high in males. Okay, so a large number of variants will be seen. All right, now since this graph is showing the number of successful matings, okay, so you see the gray graph is showing variation, right? It is showing a large number of variations. So individual 1 has this many offsprings, whereas individual 8 has only this many offspring. So it shows a large variation. Okay. So in case of polygyny, male or female, who will show a higher variation in reproductive fitness? So that will be shown by males. Okay. So that's why in this case, uh, in case of polygyny, so Q will be male and P will be female. Okay. So in polygyny, Q male and P female. Okay. Because in here, Q is the one showing the higher variation, and in polygyny, males will show higher variation in the reproductive success. Okay. So in polygyny, male is Q and female is P. Fine. Now, what will be the situation in polyandry? Just the opposite. Okay. So, in case of polyandry, it means females will have multiple partners. Now, when females have multiple partners, then females will show higher variation in their reproductive success. Okay. So, in case of polyandry, Q will be female and P will be male. Okay. So, in polyandry, Q females, right? So, Q will be female and P will be male. Okay. So, answer is option 2. This is not correct here. Yes. So moving on to another question from evening batch. So that was about Hamilton's rule. So it is talking about paper wasp. So we know that paper wasp is a eusocial organism. Okay. So the relatedness between true sisters, okay, means they share the same set of parents. So R between true sisters is 0.75. Okay. And what is given here between parent and offspring is 0.5. Okay. So genetic unit is the unit which is used for measuring R. Okay. So when a worker helps to raise four full sisters, okay, so how much R it gets, or how much genetic units it gets? So genetic units got by helping in raising one true sister is 0.75. So four sisters, genetic unit, how much it will be? 4 multiplied by 0.75. Okay, that will be equal to 3. Fine. Now, because it is taking care and raising 4 sisters, it did not do its own reproduction, right? So how much unit did it lose? Okay, so parent offspring relatedness is 0.5. So if we are talking about 4 offspring, then also it will be 4 multiplied by 0.5. So only 2 genetic unit. Okay, so taking care of 4 sisters is giving more genetic units, more benefit compared to doing their own reproduction. Okay, that's why it is evolutionary. It is like natural selection supports this kind of situation. Okay. So so according to Hamilton rule, will selection favor this altruistic behavior? Of course, yes. Why? Because 3 genetic units are gained and 2 genetic units are lost. Yes, so with that, we come to the end of this particular session where we discuss questions from Unit 3, Unit 8, and Unit 11 from morning shift as well as evening shift. And everyone, if you want to know detailed solution of this question, so very soon Biotechnica will be releasing the PDF, the answer sheet. Okay, you can go through that. And if you need even more detailed explanation of the questions, please fill up the form, contact us, and make sure that you mention the question ID so that we can answer you appropriately. Thank you, everyone. And I'm passing the control to Urmimala ma'am for the next session.
Thank you so much, Tithi ma'am. Yes, that was wonderful explanations of all the units that you have discussed from. Yes, so good evening, everyone, and I am here now with my next session. We'll be discussing about biochemistry, Unit 1, Unit 5, Development Biology, and Unit 6, that is plant physiology. Okay, so as Tithi ma'am also discussed, both the sessions will be talking about, even I will talk about. So, first we'll look at the part B questions and then the part C. Okay, now for the morning batch, morning shift part B questions, yes. Good evening, Kumar. So I hope your exams went well, all of you. Let's see. Okay, if you have any confusion in any of the question, then here you will get to know why you got it wrong. So I want you all to analyze from this discussion session. Okay, that why you got it wrong in the exam and try to learn from it. All right. So the first question was from morning session. It was based on the peptide bond formation, right? So we all know that the peptide bond is created between the amino acids of two nearby amino acids, in which we'll have the carboxyl group, right? And then we'll have the amino group, right? So these amino groups, one of the H and OH of the carboxyl group, that will be taken away to form water molecule. So that we all know it is the dehydration reaction by which the CONH bond will be formed. But here what they have asked is which molecule will be involved for the nucleophile. Okay, so that is where the question is different. It's not with the regular peptide bond concept. It is about which molecule is going to give the nucleophile. And another thing is that generally we know that peptide bond formation happens from N terminal to C terminus, isn't it? But they have said that this is a novel organism, some different organism who does the opposite. So they use ribosomes from C terminal to N terminal. Okay. So in that case, which will be the nucleophile? So first of all, you should know which is the nucleophile, right? So know that peptide bond formation is formed by the nucleophilic attack on the ester carbonyl group of peptidyl tRNA, which is bound by the P side by the alpha amino group of amino acid tRNA in the A site. Okay, so nucleotide in peptide bond is always the amine groups nitrogen. Okay, this is always a nucleophile, as this donates a lone pair of electron to form the bond. Okay, and in fact, the first step in the peptide bond formation is deprotonation of this amino group. Okay, so deprotonation leads to the form of this NH group, as you can see. So that's how the step begins, where the nucleophilic attack of the NH2 group on the ester carbonyl group and formation of the Zwitter ionic intermediate, all those will start. Again, I'm not going into detail. You have the form with you. If you feel that you need the explanation detail, you write to us and we'll give you the detailed explanation. Okay, let's focus. This session will focus why we got a question wrong. So first of all, you might think that you might not know that okay, nitrogen of the amine group is involved in that. So that might be a mistake, that you didn't read the detail of the peptide bond formation. Okay, superficially only you have read. Another thing is that you will think that since it's C terminal to N terminal, even if you knew, then probably you will think some other things. Okay, some of the other atoms. But no, what happens is that even if in reverse synthesis the peptide bond formation is a reaction, and that will remain as it is, there will be no change in it. Okay, so even in reverse synthesis, the nitrogen of the amine group will still act as a neutrophil. All right. So probably in reverse synthesis, the new amino acids would be added to the N terminal end, and ribosomes and enzymes may be structurally different to accommodate the reverse process. But for example, the peptidyl transferase center of the ribosome, the PT center, it will have to operate in a reversed manner. But the reaction of peptide bond formation will be same only, no? So this kind of things CSIR targets. Here your correct answer is option 1. Okay, moving on to the next question. This is based on the TCA cycle. So TCA cycle operates only in the presence of molecular oxygen. This is because we know that when TCA cycle, what do you get out of the TCA cycle? You will be getting NADH, right, out of the TCA cycle in various stages, in one stage, then in another stage, then in another last stage. So total 3 NADH you will be getting. In one of the stages you will be getting one FADH2, isn't it? So these are the reducing equivalents, and since it's a catabolic pathway, we will be using NAD plus, isn't it? So this is what is the function of TCA cycle. Yes, so this electrons that is given when acetyl CoA enters the TCA cycle, the electrons which are given to NADH and FADH2, they will carry it to the electron transport chain, and there, though, they will give away the electrons so that they can give back, they can come back to the form of NAD plus, isn't it? Then again they will enter this cycle. So these act as the reducing equivalents. So yes, the ultimate acceptor of the electrons in electron transport chain is oxygen. So yes, this NADH, FADH2, which are taking the electrons from acetyl CoA, they are in fact giving to oxygen as a part. So this is as it is true. Okay. So oxygen activates enzymatic dehydrogenation reaction. That is also true, because accepting of the electrons and the protons makes the NAD plus reduced to NADH and FADH2. So that is a dehydrogenation reaction. This also is happening. Correct. Oxygen removes toxic byproducts of the TCA cycle. So if you don't know, even this TCA cycle is linked to oxygen consumption also, and they will they have the production, they have the power to remove this toxic compounds also from this, from the TCA cycle. Okay, the byproducts that you have. Now oxygen activates ATP synthase. That's not quite true. So what happens over here is, even if the even this is, it activates the enzymatic dehydrogenation, so this is not correct. Okay, enzymatic dehydrogenation is happening but oxygen is not activating it, so that's why one is wrong. Second is correct. Okay, and oxygen removes the byproducts. So again, it is not dependent on the oxygen as such. Okay, it is only responsible for the TCA cycle, electron transport chain, and oxygen doesn't activate the ATP synthase. Because oxygen takes the electrons, the proton gradient is created, because of which ATP synthesis happens, but it doesn't activate it. So none of this is true. Only the second option will be correct. Okay, that's why oxygen is required. Otherwise, it will not go in the aerobic direction, it will go anaerobic direction. Third question was from the beta oxidation of fatty acids. So we know that the fatty acids, they take place in mitochondria, right? It occurs in all the, which of the following statements is true? And we know that it happens in the matrix of mitochondria, isn't it? So the first one is mentioning that it occurs in the intermembrane space, which is wrong. All the reactions are same for saturated and unsaturated fatty acids. So this is not correct, because first of all, when you see here the mitochondrial beta oxidation, it starts with the fatty acid CoA. It first of all, the fatty acids attach a CoA, and then we have alpha, beta, and before that we have the acid, the coenzyme A, that is attached with the carbon. So that is considered as the number one. Then this is number two, and this is number three. So as you can see, ultimately the oxidation step, it happens at the carbon 3. Okay, so this option 3 is right. Fatty acids, they are oxidized at carbon 3 to remove a 2 carbon unit. Okay, now oxidation of the hydroxyl group, it makes a ketone, and finally the thiolytic cleavage happens. Okay, now unsaturated fatty acids, they complicate the picture a little bit more, because they have the cis bonds, right? They have this double bonds for most, and if they are of biological origin, so they must be converted to the relevant trans intermediates which are made in step 1. So saturated, unsaturated, they don't have the same reactions, something is different. Okay, so this is not correct. And lipoprotein lipase catalyzes the first step. No, again, that is not correct, because the first step is the activation of the fatty acid. That is the first step. Okay, which is initiated by the enzyme acetyl CoA synthetase. This activation, it is catalyzed by this enzyme. So that's why this option 4 is also wrong. Here correct option is option 3. Okay, correct answer. Moving on to the next, which of the following types of interaction which of the following types of interaction will be predominantly contributing to the stability of a nucleosome, right? So what do you know about the nucleosomes? We have these histones, right? Octamer, histone, octamer is there, and around that the DNA will be there. So most of us will be knowing that the lysine residues in the histone, they are responsible for giving a positive charge, and everyone almost knows here that DNA backbone, it is negatively charged because of the phosphate groups. So negative and positive charge, the only thing which is possible is electrostatic interaction. So option 4 is correct over here. Electrostatic interactions between the DNA backbone phosphate backbone, and lysine residues of histone. This is clearly a theoretical question, that means based out of knowledge. As you can see, the DNA backbone phosphate group and the lysine residues. Lysine is a positive charged amino acid. So between positive and negative, a little bit of bonding chapter is also information is involved over here. So that gives the electrostatic interaction. Option 4 here is the right answer. Okay. Now, next, moving to the Unit 5 questions of Part B morning session. The regeneration of a severed axolotl limb. So, this is a kind of an amphibian. It is mediated by which one of the following cellular responses to injury, right? So, how is that severed means which is cut off? So, how it is regenerated? Okay, what is the processes that happen? So, if you know anything about the limb amputation, once the amputation happens slowly, we have a wound epidermis forms, then innervation will be taking place, that the nerve endings and all. Nerve supply is very important for regeneration because they release some factors. Then the apical ectodermal cap is formed, and finally it leads to the form of some blastema. So this blastema is what finally gives you the new limb patterning. Until and unless the blastema is formed, you will not see the limb patterning getting started. So that's why here the correct answer is blastema. It will grow, establish pattern, and it will differentiate into the missing link structures. Okay, so this formation of blastema is what is the critical event which leads to successful regeneration of the lost structures, and this happens through a process which is called as epimorphic regeneration. In fact, this kind of questions also they ask a lot. Okay, epimorphogenesis, the different type of regeneration. So remember, this is by epimorphic regeneration. And even if blastema appears to be unique process, but once the blastema is made, from now on it exhibits all the behaviors of the limb bud that actually formed the limb during embryogenesis. Okay. Now, second, the next question from Unit 5, Development. Very easy. I have nothing to say over here. Which is the correct hierarchy of gene activity in early Drosophila segmentation? So, we all know that positional information happens in the multinucleate Drosophila embryo. It is given by intracellular gradients that are set up by this four groups of maternal effect genes, that is bicoid, nanos, hunchback, and caudal, which then activate the next set of genes which are called as gap genes. Gap genes are called like that because if they're not present, then gaps in the embryo will be happening. They are responsible for broader section specification in the embryo. Gap genes will then trigger the pair rule genes, which will create further segments in the embryo, and segment polarity genes, they will give the polarity, that is which will be anterior posterior, they will give the polarity. So this happens in a cascade only. You have to tell the sequence. The correct sequence matches with option 2, maternal, gap, pair rule, and segment polarity. Moving on, this is from Unit 6, Part B. Which one of the following events occurs during the light reaction of photosynthesis and directly contributes to the formation of ATP? Okay, so here the trick word is directly. Okay, you might know the light reaction which happens in the chloroplast. It involves the splitting up of water molecules, which not only gives rise to oxygen but also to the protons and electrons. Now, oxygen is left out, it is released out, but the protons and electrons, they will enter the photosystem and photosystem 2, through which there will be again this electron transport chain, and ultimately those electrons will be given to NAD plus, and it will form NADPH, which will then be used in the dark reaction. So dark reaction is called because it doesn't use the light. It is where the carbon fixation happens by the enzyme Rubisco, and these NADPH and ATP, which was created through the light reaction, will be used to make the sugars, right? So anyways, this is talking about the light reaction. So option 3 will be removed off. Now option 1, 2, and 3, all of this happens in light reaction. All this happens in light reaction. Okay. In fact, because of the electron transport chain, just like oxidative phosphorylation, here photophosphorylation also involves a proton gradient formation, but here they are asking which one directly contributes. Okay, so see the direct contribution first. If you go step by step, first is the splitting of water, so that is step 1. Then is the transfer of electrons, so that will give rise to NADPH, and the transfer of electrons then will create, so that is step 2. Then the transfer of electrons, which was creates a gradient, because of which this ATP synthesis will be generated. So this is step number 3, directly after which ATP will be generated. So since the question is asking here directly contributes, that is why option 4 will be correct. Some people said that, some of our biotechnica students, they said that some statements they seemed like all are correct. So this is one example of such a question. Okay, seems like others are also correct, but you have to answer with reference to the question, not just what is generally true. Okay, now moving on to the evening shift part B questions. Again, a question from biochemistry. The pK of an amino acid chain was measured in aqueous solution. Which one of the following is the correct arrangement of the amino acids in the decreasing order of their side chain pK? So, anyways, we know that serine and threonine, they are all the polar amino acids. They have this OH group in their side chain, but they have a very high pKa. That is why at physiological pH they will remain neutral. They don't get dissociated. As such, OH is a polar group which might have the tendency to form O minus by dissociating the H plus. Okay, but serine threonine they don't do. So generally the pK for serine is beyond the 0 to 14 range. So that's why it will be considered the highest. That is why they don't dissociate within the 0 to 14 range. Okay, so serine has to be first, then lysine, arginine, histine. They are histidine. They are all the lysine, arginine, and histidine, the single letter codes. These are all the basic amino acids or the positive charge amino acid. Now, basic means they will have this amino group in them. Okay, side in the side chain. And amino group means their pKa, the point at which they will dissociate the H plus, it will be in the basic zone. So definitely it will be above 7, right? So that means lysine will come first, whose pK point is 10.5. Arginine is 12.5, it is not mentioned. Then histidine, it comes around 6. Okay. Then after that will be aspartate. So we know that aspartate and glutamate, aspartic acid and glutamic acid, they have a carboxylic group in their side chain. So that's why they are considered as acidic amino acids, right? So acidic amino acids means their pK point will be also in the acidic zone. So the glutamate pK point is, aspartic pK point is 3.7 and glutamate pK point is 4.3. So that's why aspartate will be the least, right? Here the correct answer is option 1. Very easy. Next question, again very easy. Which of the following bonds support the three intertwined polypeptide strands in the triple helix of collagen structure? Okay, so what do we know about collagen structure? It has three polypeptide chains which are called as the alpha structures, right? So as you can see, these are the three polypeptide chains, and they are primarily stabilized by hydrogen bonds. So hydrogen bond, you will know, donor and acceptor. So that's what we have here. We have the donor and then we have the acceptor. So generally when we compare with the alpha helix has hydrogen bond within the chain, so they are having intramolecular hydrogen bond, but collagen, we will say they are having intermolecular hydrogen bond. So here correct answer will be option 2. These are formed between the backbone, whatever this peptide backbone is there, no, the amide hydrogens and the carbonyl oxygen of another chain, another different chain. So they are composed of these three alpha polypeptide chains. All right. Next, skeletal muscles need to convert pyruvate to lactate while sustaining anaerobic respiration to do what? Now, what we know, when we have this glycolysis, glucose to glucose 6 phosphate and all this pyruvate, what do we generate out of this? We generate electrons. What is the entire purpose of these processes? To generate electrons, which will be given to this reducing equivalents. And after they get reduced, they will carry it to the electron transport chain, where they give their electrons and they come back to the NAD plus form so that they can enter the glycolytic cycle once again. Now, what did we just see? That TCA cycle uses oxygen. It depends on oxygen. So imagine if oxygen is not there, then TCA cycle is not going to take place, isn't it? So in that case, the pyruvate will not be going towards TCA cycle. Continuously, we will be having the glycolysis going on so that at least some energy is generated. We know from glycolysis we get 2 ATP, right? So this NADH, if glycolysis continuously goes on, NADH continuously needs to be produced. For that, NAD plus is required. But NADH and NAD, they are in the same equilibrium pool, isn't it? So NADH will not be giving the electrons to electron transport chain. It has to be recycled back to NAD plus. So that is why we have this anaerobic reaction where pyruvate gives the electrons. Pyruvate accepts those electrons from NADH. See, in somewhere they have given the electrons and formed pyruvate, but now the reverse will be taking place. Pyruvate again takes back the electrons and converts into NAD plus, so that it can start the glycolysis again. They can enter into the glycolysis. So it is just to replenish the NAD plus for glycolysis to continue. So correct answer is option 3. If you feel confused in this part, so this is a part of a broader topic. This entire metabolism of carbohydrates is a related topic. Until and unless you learn it in a proper way, it will be difficult for you to grasp. So the form is already given down. If anyone wants detailed explanation of any of the questions, Biotechnica is here to help. Do fill in the form and we will get back to you. Okay. Then coming to Unit 5 morning session, Part B questions. In a developing Drosophila embryo, which one of the following is the correct order of Hox gene expression from anterior to posterior? So we know that Hox genes, they are expressed along the anterior to posterior way. This is anterior to posterior, and they are expressed in a specific order. Okay, so we have this entire thing. This is one complex, Antinapedia complex, and then we have the ultrabithorax complex. So they have just asked about three genes: Antinapedia, ultrabithorax, and abdominal A. So if you see here, Antinapedia, ultrabithorax, abdominal A, these three are in a sequence. What are Hox genes for? They're responsible for forming the body structures in a certain area, right? And Hox genes are activated by all those what we saw before: maternal activity, maternal effect genes, gap genes, parul genes, and ultimately they will be activated. As you can see, not all the Hox genes are activated in all the areas. So whichever region has whatever Hox genes, like Antinapedia is expressed in the T2 section of thorax, where a pair of wings will be coming out. So Antinapedia is responsible for that structure formation. Okay. Next question is which one of the following from Unit 6? So I just want to say that few of the Unit 6 questions will be discussed in the next session. So stay tuned. They will be discussed by Divya ma'am. And some more questions will be discussed in the later sessions. We will be having the other units on 19th. So another day we will have the sessions. Just wanted to remind you that there will be a Kickstarter session on 17th March also, Monday. So don't forget to attend it for the preparation of the next CSIR exam. So this question, excuse me, this question is for which of the following statement represents correct sequence of events during electron chain from P680 to P700 in the light reaction. So I told you that the light reaction, water will be split up and electrons will be released. So these electrons will be transported from the photosystem. So this is what the photosystem you have. The PS2 is P680, which means it accepts light in the 680 zone, and P700 it accepts in the 700 nanometer wavelength zone, right? So once these electrons, they will take the wavelength of light, they get excited. These are the excited chlorophyll molecules, and after that they pass the electrons to the carriers one by one. Okay, they will pass it to P700, and P700 further does it forward. Okay, so which are the electron acceptors that we have here? P680 to pheophytine, that is in option 3 it is given. Then plastoquinone A, plastoquinone B, and then we have the plastoquinone pool B we have. So after that directly it is passed to cytochrome B6F complex, and finally to plastocyanine, then it will be given to P700. Okay, pretty straightforward question. Have this type of questions have come in the past as well. Okay. And then another question regarding the herbicides. So these type of questions are also very common. The acetohydroxy acid synthase, which is called as the AHAS, this is an enzyme which is involved in the branched chain amino acid synthesis. Okay, so like valine, leucine, isoleucine, and it is inhibited by all the following classes of herbicides except. So AHAS is a very common target site of total 5 AHAS inhibiting herbicide chemistries. Okay, so those are the sulfonylurea SU category, then imidazolinone, which is the IMI category, then we have the triazolopyrimidine category, pyrimidinil thiobenzoates, and sulfonyl aminocarbonyl triazolinol. Okay, so that is another kind of a category. So total 5 categories will be there. As you can see here, we have all the 3 which is already given, right? triazolopyrimidines, then sulfonylureas, and imidazolinones, except for L-phosphinothrycin. So this also known as glufosinate, if you have heard before, L-phosphinothrycin, which is known as glufosinate, it inhibits a different enzyme. It doesn't inhibit this enzyme. Okay, so this is the other enzyme it will inhibit is glutamine synthase. Okay, GS, which is involved in the glutamine biosynthesis. So this is also pretty straightforward. It's just that you have to study these different herbicides. Okay, moving on. Yes, the fear point of everyone, the part C questions. Let's have a look at the morning shift first. So morning shift saw the questions. First question was on enzymes, right? Enzyme has been found to efficiently catalyze the following reaction as is given. So A converts into B, B converts into A. The rate is forward rate reaction, rate of forward reaction, how fast it will go, and rate of backward reaction. And equilibrium constant is given by forward by reverse. So we have already got it. Now whoever will be knowing little bit about enzymes, you will know that the enzymes do not affect the equilibrium position. K equilibrium, it depends on the free energy of the substrate and the products. Whoever's free energy is lesser, that side will be preferred, and that part, whether it's a substrate or product, accordingly that will be preferred. Okay, enzymes do not disturb the equilibrium, so neither keq nor 1 by keq can be answer. They have asked that what will be increased if you use enzymes for the uncatalyzed reaction. So they are not asking what will happen in uncatalyzed. Obviously, if a reaction is uncatalyzed, means this rate will be very, very slow, isn't it? Kf also will be slow, and Kr will also will be slow. Only if you increase enzymes, then only this apply enzymes, then only the forward and the reverse rate will increase, right? You might think why reverse rate will increase. So you should know that enzymes accelerate both the forward and reverse reactions equally. This is a concept which all might not know. They will accelerate if they are increasing. If you use enzymes over here now, okay, then they will increase the kf also, and they will increase kr also. Okay, so that is why option 1 is the right answer here, right? Because if kr is increasing, then 1 by kr will be decreasing. So they have asked which is increasing, so answer will be answer will be kr, option 1. Kf is not there in the option, so no question, right? So this is a conceptual question. Generally part C is conceptual. This was a very interesting question. 3 strands of a beta sheet of 4 peptides. So basically I'll tell you what is this question about. We have 4 peptides, okay, and we have these beta sheets. You know, these are the strands in a beta sheet. So what is the concept that is required over here? um, when you have a parallel beta sheet, so let's say this is parallel beta sheet, okay, and when you have an anti-parallel beta sheet, see n to c, if you start, then to form anti-parallel you will come back like this only. So this forms a hairpin loop like structure, so that you will have this c, again n to c, so the opposite ends don't match. That's how it is anti-parallel. But if you have parallel, this will go like this. It has to take a hairpin loop and then it has to come back. So hairpin loop means, sorry, not hairpin loop, it will take a complete 360-degree loop. Okay, it's not a hairpin turn, it's a 360-degree loop it will take. So that will involve more length of the peptide. That is not part of this part, this region is not part of the beta sheet. Okay, but if you have parallel arrangement, then always they have to come 360-degree and then only go in the correct direction, right? So basically the concept involves that for anti-parallel arrangement, you will require less connecting length of peptide, and for parallel arrangement, you will need more connecting length of peptide. So they are asking which one of the following option is correct regarding lengths of this peptide. Peptide 1, 2, 3, and 4. Okay, so they have mentioned that 3 strands of this peptide are linked by amino acid sequences of the smallest length possible. So why this they have said the smallest length possible, right? So smallest length means when you go for like this, okay, let's say parallel arrangement, you start like this, okay, it might come back like this as well, the polypeptide chain, but it's also possible that here they will take some other loops and turns, okay, which are not possible to be in the beta sheet. So that can be an infinite loop, okay, infinite length. So they have mentioned that there are no such other unnecessary structure. It's a direct contact. So now you will see for peptide bonds, Neha, I'll just get back to you. Just let me finish this question. So here we have this anti-parallel arrangement, right? So from here we will connect like this, okay, in peptide 1, and again this is anti-parallel, so again like this, right? So in both we have used shorter connecting lengths. In peptide 2 all are parallel. So from here N to C, we'll start from here, then again take a loop, okay, then again take a loop, then come back. So this is what is the longest one, right? And this is the shortest one. Now peptide 3 and 4, they are almost same length. Why? Because when it starts from here, you will be requiring one 360-degree loop and one beta turn, right? And here you will first need a beta turn and then again a 360-degree loop. So more or less these are the same length only. So that's why here option 2 is right. Peptide 1 will be smallest because it has all the antiparallel arrangement. So shortest sequence, then will be 3 and 4 both, and then will be peptide 2. Option 2 is the right answer. Yes, how many of you got this question correct? Did you attempt? Yes, Neha. So next batch, it's about to start. It's your lucky day. Yes. Yeah, we have the Shaurya batch, Sneha, and all others who are interested in joining the next CSIR NET batch for the upcoming CSIR NET exam. That will happen mostly in July or August or September. We don't know. We just had the exam now. So I'm guessing it won't be June, but it may happen in August or maybe in September. Let's see. We can write to CSI to conduct it before itself, right? So Shaurya batch will be starting soon, and 20th March onwards is the date. We'll be sending you notifications for that. You can contact our team. You have the contact details, and most importantly, you fill up the form over here. And there also we'll be getting back to you if you fill up the form. Then if you have any doubt in the question, and we can get back to you. Okay, Sneha. All right, moving on to the next part C question in morning session from Unit 1. At what range of substrate concentration will an enzyme with a kcat of 30 second? So we know here kcat is like we know it's a turnover number, but let's see whether we require it or not. And a km of 0.005 molar show 1/4 of its maximum rate. So it's just showing that how will it like you want to find out at which substrate the velocity will be 1/2 of its maximum rate if the km is this much. Okay, so first of all, we know the Michaelis-Menten equation. We have the initial velocity is equal to Vmax into S substrate, maximum velocity into substrate, divided by Km plus substrate, isn't it? Now here, the enzyme needs to work at 1/4 rate of its Vmax. So how can we write it? V0 is equal to 1/4 of Vmax. Or simply we can write V0 is equal to Vmax by 4. That's it. This we have to replace it here. Instead of V0, we will be writing Vmax by 4. Okay. Vmax by 4 is equal to Vmax into S divided by Km plus S. Okay. So here Vmax is in the numerator. So this gets cancelled. Then do cross multiplication. So Km plus S will give you 4S. Okay. Now 4S, S you bring it to the other side. So you will get Km is equal to 3S, right? So they have asked at what substrate. So if you have to take out the substrate, it will be Km by 3. Already they have given the value of Km. So that is 0.005 divided by 3. Okay, so that will give you somewhere around 0.00167 molar, right? So from here, if you want to make it in the form which they have given, so 1 point you have to shift the points 3 places. So 1.67 into 10 to the power minus 3 molar. So that's why option 3 is the right answer. Okay, pure numerical based question. KCAT they have simply given to divert you, not needed at all in this question. All right. Next is also from biochemistry only. A cytoplasmic monomeric protein containing a single non-surface exposed cysteine residue precipitates upon mutation of the cysteine to isoleucine. So single non-surface exposed. Non-surface means inside. It might be involved in some kind of interaction. Okay. But when it has been moved to isoleucine. So what do you know about isoleucine? It is a branched amino acid, right? And cysteine, what do you know about it? It's a small amino acid which has this SH group in the side chain. And you know SH group can be involved in interactions, hydrogen bond or disulfide linkages. So probably it was stabilizing the protein inside, but when it is converted into a linear, that is, they are the aliphatic branched amino acid. So when you convert it into a linear amino acid, probably it will be broken, right? It is the connection will be broken, and this branched aliphatic amino acid may be a hindrance in the center of the protein because it is non-surface, means it is towards the core. So inside the core where there is a restricted space, they might be hindering over there, right? So that's why option 4 seems to be correct. Mutation, cysteine mutated to isoleucine causes steric hindrance in the protein, while mutation to alanine doesn't. Why mutation to alanine does not create any problem? It again leads to a soluble and non-functional protein. So it folds in the proper way and it interacts with water also properly. The only way it precipitates is because isoleucine hinders the folding in the center, so it turns inside out and then it cannot interact with water, it moves away from the water, right? It precipitates. But alanine, comparatively, it is also a smaller amino acid, same like cysteine, so that is why converting into alanine doesn't create any problem. And anyways, alanine also is hydrophobic, so it will happily enjoy in the core of the protein because core of the protein is hydrophobic in nature. Concept based question. If you know about the knowledge of amino acids, then you could have solved this. Another numerical question in morning batch, morning session shift from thermodynamics. Okay, so here they have given you a reaction and how much substrate and product concentration is there, glucose 1 phosphate to glucose 6 phosphate. They have mentioned at equilibrium. Now, at equilibrium means what they have asked you to find out, the KEQ value and the delta G. So KEQ is what? What can be simple? Nothing can be simpler than that. KEQ is concentration of product divided by concentration of substrate, isn't it? So they have given over here 9.6 into 10 to the power minus 2 divided by 4.5 into 10 to the power minus 3. So that will give you around 21.33. Okay, so that is the KEQ value. Now delta G0 means what? We know that delta G0 is equal to minus, it is minus RT ln KEQ, isn't it? So what is R? It is the gas constant. It is 8.314 joule per mole dot Kelvin. I'm not writing the units. Temperature here they have mentioned 25 degree. So 25 plus 273, you have to always express in Kelvin for this calculation. 298. ln value will be 2.303 into log. So through 2.303 into log of 21.33, which is the KEQ. Don't forget there is a minus over here. Okay, so if you solve this entire thing, you will get somewhere around 7531.8, but you will get this in joule per mole dot Kelvin because you have taken R value in joule per mole dot Kelvin. So shift it to three places, shift the point to three places, 1 2 3. So minus 7.5 into 10 to the power 3, so 3 joules per mole dot Kelvin, and 10 to the power 3 joules means kilojoule per mole dot Kelvin. So minus 7.5. So that's why option 2 is the right answer. Okay, again simple question, just that you have to be little bit alert about what you are doing. Okay, now coming another biochemistry question, ABZ DNA. So here we have given below two different structural features, two columns, and DNA RNA conformations. So do I really need to tell you this? Left-handed is Z form. We all know, right? So we have the A and B DNA. See, the top segment is going towards the right direction. So that means it's a right-handed structure. B is also a right-handed structure. Z, you will see it going the other way. So that's why it's a Z. It's a left-handed. So A will be 3. A will be 3 means either 4 or 1. Now number of base pairs per turn is 10. So again we know that B form is the normal Watson-Crick model, which will have 10 bases, 10 or 10.4 to be precise. Okay, so B will be 2. So yes, that's it. Nothing else is required. Such an easy question, 4 marks. Can you imagine? Yes, it's literally given free of cost. The base pairs are off-centered in A form. So off-centered means what? The base pairs, they will pass through the center in case of B DNA. But as you know, A form is the dehydrated form, which means it will be more shorter and wider, right? Wider means the base pairs will be kind of shifted towards the peripheral zone from the center. So they will not pass through the center. That is why it is off-center. So they will be base C will be 1, and RNA, it cannot be in the form of D form. So it will be only in the form of A form only. It can be Z form also, but double helix means mostly it is A form only. Because RNA has this oxygen. Remember, in DNA, you have at the 2 dashed position, it's a deoxy at the 2 dashed position. But for RNA, we have oxygen at the 2 dashed position. And that creates a steric hindrance when it comes in the B form. Okay. So that's why here option 1 is the right answer. Coming to the unit 5 questions of morning batch. So they have given some development processes and their associated molecules or pathways. So one is the dorsal ventral axis in amphibian embryo. So if anyone would have studied this before, you all know that amphibian embryo, the dorsal side, we have the WNT beta catenin pathway, and the ventral side, we have the BMP signaling. So BMP is the ventralizing signal, and WNT is the dorsalizing signal. And whichever side we have dorsal, we will get this nodal also activated. And in case of mammalian limb development, if you have the dorsal and ventral, that is done by the WNT7A. Whichever side of the limb gets WNT7A activation, they will activate the LMX1B. So that side will get made into dorsal. And whichever side, ventral side, we will have the engrailed, wherever it will give the signal, that side it will become the ventral side. So BMP actually activates this engrailed one. So that's why here option A and D are correctly matched. Okay, it's not match the following, it's just associated terms, are they given properly or not. Okay, sorry, not A and B, not A and D, A and B. So dorsal ventral axis specification, engrailed WNT BMP, and dorsal ventral and amphibian WNT BMP for activin or nodal. Now, when you talk about Drosophila dorsal ventral axis, there was a question regarding this. I'll discuss. So it doesn't depend on this FGF. It depends on this gherkin and torpedo. It's TGF-like factor. Okay, so this is not correctly said. An anterior posterior axis formation in mammalian limb. So that is given by ZPA. Okay, so ZPA is not mentioned over here, and that too it is TGF factor beta. What is involved over here, right? So that's why here option A and B, option 1 is the right answer. Moving on, this is a question again from unit 5 only. RAB5C and diamine. So these two molecules are there. First of all, they have said that FGF8. Now FGF8, it forms a gradient. Gradient means what? From wherever the higher concentration, it will become the lower concentration. That's what is gradient, right? So whenever in an embryo, wherever the source will be there, the concentration will be high, right? So it depends on the gradient formation, diffusion from a localized source. So from this localized source, it will diffuse. That's how the gradient will form. And it also depends upon removal of the FGF8 through endocytosis. So wherever the low concentration has to be there, little FGF will be released, so that it also contributes to gradient formation. Okay. Now both RAB5C and dynamine, not in a together way. Okay, it is not like it synergistic way. Both will be required. Not that. It is both separately. They will help endocytosis. Means RAB5C can also promote endocytosis, and dynamine can also promote endocytosis. So what would happen to the FGF concentration gradient when RAB5C is overexpressed or dynamine is inhibited? Okay, so overexpressed means what? There will be more removal of the FGF8, right? If this RAB5C is overexpressed, then more FGF8 will be moving out. So what can you expect here? You can expect this dashed line is what is RAB5C. You will expect the gradient to be little bit steeper, right? Because from the further areas, more and more endocytosis will happen because it is overexpressed. Okay, so in case RAB5C is overexpressed, you will see a more steeper gradient as is given over here. and dynamine if it is inhibited. Now dynamine is supposed to remove the FGF8. Now imagine dynamine is not working. Then in that what will happen? This removal will not happen properly. In that case, it will be a more flatter gradient, okay, than the normal one. So if this is the normal one, which is the solid line, then dynamine will be little bit more flatter gradient that will be created. So that's why correct option is option C. Nothing to understand, it's about analyzing the question only. Embryos of a species display conditional specification at 16 cell. So here itself they have given you the trigger that it is conditional. Conditional means what? If you have a group of cells, okay, and you transplant some cells over here, then this whatever cell you are transplanting here, if it is conditional specification, then they will forget what they were making and they will make the cell which in which it is transplanted. So here you have the vegetal blastomeres which is supposed to give rise to endoderm, right? And the animal blastomeres which is supposed to form the ectoderm, right? So here the vegetal blastomere, from here it is taken and transplanted into the animal blastomere. So now remember, which is the transplanted tissue? The grafted cell. What is the grafted cell? They have asked what is the outcome for grafted cell. So the grafted cell here is the vegetal cell, isn't it? Which was supposed to become endoderm. Supposed to. But you have to focus on what they have previously mentioned. It is conditional specification. That means when it is transplanted over here, along with the animal cells, which are supposed to form ectoderm, so this graft will forget to make endoderm and it will also make ectoderm. Okay, so that's why here option 4 is the right answer. It develops into ectoderm, right? It is only hint over here is conditional specification. If anyone got it wrong, then now you know why. Some students they might have applied a knowledge of sea urchin transplantation. So generally we study this in our classes. So there also the vegetal blast micromeres are transplanted to the animal pole, because of which they start a secondary axis. But here, neither they have mentioned sea urchin, so embryo of a species, and nor have they said that some organizer properties they will be having. Any other information we don't have. So from we have to solve from the options only by what is mentioned in the question and not what knowledge we generally know. I always say this in my classes. That for development biology, any experimental type question, don't forget about what you know. You have to only apply based on the question what they are asking. Okay, we have the form below. We have already given you the link. If anyone wants detailed explanation of the questions and you have you think that no you feel in a different way, or you have solved it this way, was it correct or not, you can write to us. Okay, we'll get back to you. We teachers are here to help you. And this question also from morning session, it is from plant only. So this question was very interesting. You can see here, again you see this enzyme came into picture, AHAS, which was involved in the amino acid, branched chain amino acid synthesis, right? So here this AHAS is making this oxoisovalerate. Oxoisovalerate will be, pyruvate is converted to oxoisovalerate, okay, by this enzyme AHAS. Oxoisovalerate, partly it is making leucine and partly it is making valine. Okay, partly leucine and partly valine. Now, this leucine, if it is too much, then it goes for a feedback inhibition. Okay, and by the way, oxoisovalerate, if it has to be converted into leucine, then this enzyme will be required. Okay, so if there is too much of leucine production, then it goes and feedback inhibits its own synthesis. On the other hand, threonine will be made into isoleucine by an enzyme TD, and if isoleucine is too much, then it goes for a feedback inhibition for TD. But in case valine is present, then valine will relax this inhibition. That means if valine is high, then it will not allow isoleucine to go and inhibit its own synthesis. So that's what happens over here. They have asked which of the following possibilities of these pools, branched chain amino acid pools, is likely to occur in the RNAi knockdown of IPMS. So RNAi knockdown means IPMS is not there. Simply IPMS is not there. So now if IPMS is not there, I told you oxoisovalerate was making leucine and valine. just because of this. If this is not there, then entire of oxoisovalerate will go for valine production. Okay, that means valine will increase. If valine increases, then it stops the feedback inhibition from isoleucine. So isoleucine production will not stop. Okay. Generally, if it was more, it would have stopped its own production. But because now valine production is increased, it will stop this isoleucine feedback, because of which isoleucine will keep on getting produced and its concentration will increase. So both valine and isoleucine concentration will increase, whereas the leucine, it will not be made only, right? Its concentration will decrease because IPMS you have blocked. Again, no previous knowledge required. It's just that you have to see the flowchart and you have to analyze. A similar question, a more twisted one. This is a simpler form. A more twisted form has been given in the evening shift. So since it's a similar one, I won't discuss it because of lack of time also. So quickly, I will just discuss few of the evening shift part C questions from unit one, that is biochemistry. This question was ribose sugar pucker in nucleic acids, which are labeled A, B, C, D with the corresponding conformational states. So this is the base. Now, if you know anything about the ribose sugar, generally we will write like this, right? But this is, and then the base will be connected. But this is the puckering form because of the tetrahedral nature of carbon atom, right? So you can see this is the plane, okay? Actually, endo and exo, they are two different things. Endo means if you consider the plane of the molecule, then if any atom is lifted up above the plane, it will be called as endo, whichever atom that is. And if it is down of the plane, it will be called as exo. Okay, so we have different conformations like this, different sugar puckering we have. So the A1 form, it is showing that the 2 dash endo, see the base will be connected to the C1 and this is 2 dash endo. So A will be 2 dash endo clearly. 2 dash endo means either 2 or 3. Now have a look at C. So here you will see that the 2 dashed is outside the plane, out of the plane. So it will be 2 dashed exo, but 2 dashed exo is not mentioned over here. Let's look at the 3 dash now. So C3 dashed is inside, that is endo. So that's why C3 dash endo, option 2 is the right answer. Okay. Now B over here, it is talking about O4. So C5 means this will be C4 and C4 means the O connected with it, that is the O4. So that's why this is on top of the plane, above the plane. So that's why it is O4 dash endo. On the other hand, if we take here C5 dashed it on top, that means this is C4 dashed and the oxygen is pointing below the plane. So that's why here it is O4 dash exo. So they have just given in general, they have not asked A, B or Z DNA, but sometimes these questions also come with respect to A DNA or B DNA. So in A DNA we have C3 dash endo and in B DNA we have C2 dash endo. So that's the difference. I was telling you that RNA cannot have the B DNA form. The reason is because 2 will have oxygen and with the endo conformation it will have steric hindrance. Okay. The next one is on line Weaver Burke plot. Evening shift one question was there. So if you know this line Weaver Burke plot, first of all, this graph, it is mentioning the solid line is without inhibition and the dash line is with inhibition. Okay, without and with an inhibitor. So we can see here, this point is the y-intercept where the graph cuts the y-axis. So this is what is called as 1 by Vmax. We can see in both the graphs 1 by Vmax is not changing, which means Vmax also will not change. And when does Vmax doesn't change? In case of competitive inhibitor. Okay, so by that we know it's a competitive inhibitor. And this x-intercept, it is given in a Lineweaver-Burke plot, it is given by minus 1 by km. Okay, so here this is the one without inhibitor, which is minus 1 by km, and this is the one with inhibitor. So we can see it moving towards 0. Even here also we can see moving towards 0 from minus 3 to minus 1. So that means it will increase over here. Okay, so km basically increases. Why km increases? If 1 by minus 1 by km is increasing, that is moving towards 0, it means minus km will be decreasing, and minus km decreasing means km will be increasing. So we know that km is increasing, right? So what we'll write here from the x-intercept, I'll remove this part. So from the x-intercept, we know that the x-intercept is minus 1 by km, right? And if you talk about the one without the inhibition, so here the value is minus 3. So minus minus gets cancelled. That means km will be going up here. That means what is the value of km if you take it up here? So km will be 1 by 3. This is the value of km. Now let's look at the one with inhibition. So with inhibition, we will have minus 1 by alpha km. Alpha factor is the factor if you have the inhibition. Okay, minus alpha factor, minus 1 by alpha km will be equal to minus 1. Okay, because here it is shifted to minus 1. Again minus minus cancelled, so alpha km will be shifting over here. That means alpha km is equal to 1, right? So here we have already calculated km is 1 by 3, right? So if we can substitute the km over here, alpha into 1 by 3 is equal to 1. So 3 goes over here, which means alpha is equal to 3. We know now the alpha factor is 3 over here. So if you know something about the competitive inhibition, alpha factor, it is given by 1 plus inhibitor concentration divided by the Ki, which tells you regarding the affinity of the enzyme and the inhibitor. So again, if you want a detail, I'll send you later. Here alpha factor we have just calculated, it is 3, and then 1 plus you have concentration of inhibitor. So inhibitor concentration they have already given, that is 1, and then we have Ki value. So Ki value you need to find out. So this one we can put Ki and then Ki plus 1, right? So Ki will bring it here. So it becomes 3 Ki is equal to Ki plus 1. Okay. So Ki will come this side, which means it will become 2 Ki is equal to 1. That means Ki will be 1 by 2, which means 0.5 is the right answer. Okay. It's a graphical analysis plus the numerical problem solving also skills. Coming to the unit 5 questions of uh, evening shift part C questions. So WNT beta catenin signaling plays important roles. So if anyone knows about the WNT signaling, in the absence of WNT signaling, there is no WNT signal on frizzled. So frizzled is not activated, disheveled is not activated, because of which this entire axine complex, GSK APC axine complex, is formed in absence. Okay, and that is why it will phosphorylate the beta catenin. It will bind the beta catenin and then it will ubiquitinate it and degrade it also. Okay, so that means beta catenin cannot enter the nucleus, right? But in the presence of beta catenin, so that's why A will be correct. In the absence of beta catenin, in the absence of WNT ligands, beta catenin is phosphorylated by this complex, which leads to its degradation. Then if you have beta catenin WNT bonding, frizzled is activated, DSH is activated, and DSH will inhibit this GSK complex formation, because of which beta catenin is free. It is not anymore bonded and it cannot be phosphorylated. It is free to enter the nucleus and trigger the transcription. So it is said that beta catenin TCF complex acts as a repressor. No, repressor is not correct. It acts as an activator of gene expression upon activation. Also, the WNT catenin is initiated by binding to WNT ligands to receptor tyrosine kinase. No, frizzled is not receptor tyrosine kinase. It's a separate category of receptor. So C and B is wrong. And D, beta catenin is not only involved in this transcription regulation, but as you can see, these are cadherins which are involved in cell to cell interaction. Two cell membranes are given. And the cytoplasmic site of the cadherin, we have this alpha catenin, beta catenin. So they also help in cell to cell addition as well, not just transcription regulation. So option 3 A and D is the right answer. A pure theoretical knowledge question. Next is regarding the complement system. So it is asking about the alternate pathway. If you know that alternate pathway, as in the lectin and the classical pathway, it doesn't require these antibodies binding to pathogen. Okay, so it is saying that it is initiated when antibodies bind to pathogen. This is not correct. Then pathway is initiated by spontaneous hydrolysis of serum complement. So this part is correct again. B option is correct because C3 anyways will be cleaved. So that is how the complement system will be started. But as you can see, the pathway uses different C3 and C5 convertases. So C3 convertase in case of classical and lectin is C4B2B. But in case of alternative, it is C3B5, B6 created from different fragments. and even the C5 also, because they are combining with different fragments, so C5 convertase also will be different. So here they are saying same, that's why it is wrong. A and C is wrong. D is again correct because alternative pathway is in fact triggered by properdine and the factor D, okay, which is the prothrombin. So here correct option is option 3. All right. Then we have the unit 5 question, which is the figure based on the morphogen. So morphogen here we know that it's from the source it will be high concentration, and to the end of the source it will be low concentration. You can see how the different cells are getting different concentration of the morphogens, isn't it? So here the concept is that if these cells are receiving high concentration of morphogen, then even if they have less affinity, then also the effect will be there. But the outer cells, they have low morphogen gradient, so their affinity should be high. So that's the concept that they have used over here. Okay, so you can see that this threshold means beyond that a different cell type will be formed. So this cell type 1 is activating the expression of ABC genes. Okay. Cell type 2, DF it is activating, but B is present in cell type 2 also. That means B is activated in cell type 1 and 2 both. So clearly this B will not have same affinity. So 3 will be wrong. Same affinity, morphogen will not have same affinity for A and B. It can have for A but not for B, because B is expressed in both the types. Okay. Also it is said that the transcription, it is said that which one will not be which will be correct over here. Now if you see F is expressed in cell type B, cell type 2, but it is C is expressed in cell type 1. That means F will be having higher affinity because F will come in a lower gradient, C is coming in a higher gradient, so its affinity can be less. So F would have higher affinity than C, so option 2 is correct. Now, A and D, if you take, D is expressed in cell type 2, whereas A is expressed in cell type 1. That means D should be having higher affinity. But here it is saying higher affinity for A. That means this is wrong. Okay. And M, if you compare M and C, so M is in third one, which should have the highest affinity. But it is saying low affinity than that of C. So that's why 4 is also wrong. Here the correct answer will be option 2. Okay. Now, this is regarding the dorsal ventral axis formation. So, this patterning in the oocyte depends on the expression of gherkin. I am not going into the detail of this question. It's just asking what will happen if the maternal deficiency of gherkin is there. Simple thing you know, gherkin, whichever side signaling will be there, that side you will get dorsal side. With gherkin signaling, you will get dorsal side, and the other side you don't have the signaling, because of which dorsal is free, it enters the nucleus, whatever it is, I'm not going into detail, it will give rise to ventralization. So if maternal deficiency of gherkin is there, that means dorsal side will not take place. That means dorsal will also form ventral side. It will lead to the ventralization of the embryo. The link is below. If anyone wants a detailed explanation, you can message me. I will skip this question also because of lack of time. um, yeah, so this question you can see, if planaria is cut in reverse. So planaria, it what happens, this WNT signaling is very important for specifying the posterior identity, that is tail formation. Okay, so here they have said if the ABC is overexpressed in the head piece, it will block the tail regeneration. So that means from the head piece that towards the here, if you have the overexpression of ABC, then it suggests that ABC might act as an inhibitor of the WNT signaling, because to make the tail and all, WNT signaling will be required. But here if you overexpress ABC, then the tail regeneration, it is not happening. That means this might be an inhibitor of beta, that is WNT. Okay, so ABC inhibits WNT, and WNT will be activating beta catenin, that we know. So it will go for tail regeneration. Okay, but it is also saying that when constitutively active beta catenin is there, it is overexpressed along with ABC, still tail formation will occur. So that's fine, because constitutively active beta catenin means even if ABC is inhibiting WNT signaling, but beta catenin is still active. Okay, even without the signaling, beta catenin is active. That is what is constitutive active, it means. So even without beta catenin, if it is active, then it will not stop the tail regeneration. It will still happen. So ABC functions as a negative regulator of the WNT catenin pathway, and WNT and beta catenin is the key effector promoting the tail regeneration. Okay. So last question, it is based on this plant interconversion of fructose to fructose 1,6 bisphosphate. It is a critical step in central metabolism in plants, and these are some of the statements regarding this interconversion. Phosphofructokinase catalyzes the C6 phosphorylation of fructose 6 phosphate. So clearly it is not C6 because fructose 6 phosphate makes fructose 1,6 bisphosphate. So that means it is adding the phosphate in the first position, not C6. A is not correct. Then plastid phosphofructokinase is activated by PI, while cytosolic is activated by phosphoenol pyruvate. Again, this is not correct because this plastid phosphofructokinase, it is inhibited by plastid one is inhibited by phosphoenol pyruvate and ATP, and it is activated by phosphate. Okay, so it is inhibited by these two. So here it is saying activated by PI, it is saying the other way around. And cytosolic enzyme is principally inhibited by 3-phosphoglyceric acid. And this inhibition is only partly relieved by phosphate. So that's why B is wrong. Cytosolic fructose 1,6 phosphate is strongly inhibited by fructose 2,6 bisphosphate. So you can see here, this is the cytosolic fructose 1,6 bisphosphatase enzyme, which converts the fructose 1,6 bisphosphatase into fructose 6 phosphate. So here also, this fructose 2,6 bisphosphate strongly inhibits it, and that's why C is correct. And pyrophosphate dependent phosphofructokinase, it catalyzes a reversible reaction of interconversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Okay, so we know that fructose to 1,6 bisphosphate is generally an irreversible reaction, but if that is the regular PFK1, but if you have the pyrophosphate dependent phosphofructokinase, it is a different category which gives a reversible reaction. Okay, so this one question I have missed it out. It's regarding the E-cadherin and the hippo pathway. If you want the explanation, you can let me know. The principal solution of this is that the trophoectoderm and the inner cell mass will be expressing the OCT4, and the trophoectoderm layer in mammalian, it will be expressing the CDX2. So the CDX2 expression will be triggered by the hippo pathway. Okay, because of which it will activate the MST and the LATs. Okay, these all it will activate, and MST LAT will phosphorylate and inhibit the YAP and the TAZ. So that means if you have this hippo pathway, then YAP TAZ will be inhibited. Okay, so that's what happens in the inner cell mass where hippo pathway is there, but in the trophoectoderm hippo pathway is not there. Okay, so cadherin, which is triggering the hippo pathway, the inner cell mass cells are connected with each other by cadherin, as you can see. So they will trigger the hippo pathway, because of which the YAP and TAZ, they are inactivated. They can't enter the nucleus. If you see in the trophoectoderm, they were able to enter the nucleus. Here they cannot enter the nucleus, and that is why they cannot trigger the production of CDX2, which was happening in the trophoectoderm. Okay, but in the inner cell mass, they can't make CDX2. So that is why OCT4 is not inhibited. Generally, CDX2 inhibits OCT4. But here, OCT4 will be maintained or synthesized. If OCT4 is maintained, it goes for pluripotency. So that's why I think this option should be option 3 answer, because here it is said that in inner cell mass, the YAP and TAZ is activated. So if they are activated, how can the OCT4 expression be there? That doesn't make sense. Okay. Instead, because of the E-cadherin, hippo pathway will be there, because of which you may have active MST LATs. Then only you will get the OCT4 expression. So I'll have to check for this source. Then in case any one student wants to challenge this, this might be a wrong option selected by CSIR answer key. So that's all from my end. Thank you everyone. And I hand over the controls to Divya ma'am now.
Hello everyone. Good evening to all of you. I hope so you are able to see the screen. I hope so you are able to see the screen and you are able to hear me properly, right? So thank you, Urmi ma'am, for that. And I hope so you got a very insightful webinar going on right now, because just now only the answer key has been released, and we are trying to figure out like which question is more twisted and which question is easily given. So like that, we are trying to figure out from the past two sessions. So this session, that means the third session, we will be covering the questions from Unit 2, Unit 4, Unit 6 and Unit 12. Good evening, Kumar. So last time, the CSIR NET exam, you had a huge number of questions coming from Unit 12, and that too, it was like very tough question, which was combined with genetics, techniques and all. But this time it was like, as always, we used to get very less number of questions, hardly one or maybe two, like that we got. Compared to other units, yeah, they are maintaining their frequency, and the important topics which we always consider, from there only we have got the questions. So let's start with our questions. So first I'll be covering up the morning shift, and then we will move on to the evening shift. So in the morning shift, you'll find the very first question was regarding the modification that happens in the histone molecule. Anyone who is having a very basic idea about the chromatin organization will definitely get into an answer. So they are asking where exactly the modification takes place. So the modification takes place specifically in the N-terminal region. Reason? Because of their packaging. If you see the tetramer, the dimer, after the DNA has been covered, you will find that only the N-terminal tails are exposed to outside. So these tails will be susceptible to all kind of modifications. So whether you go for phosphorylation, whether you go for methylation, acetylation, ubiquitination, everything happens majorly in the N-terminal region, with some exceptional cases, as you can see over here, in case of H3 and H4, where it happens in the histone fold domain. But still, majority of them will be taken place in the N-terminal region. So this was a very easy question, and I hope that everyone should have attempted this. Moving to the second question, another easy question. This time actually a lot of easy and direct questions you got in the CSIR NET exam. So this is regarding the solute transport, how the molecules are moving across the phospholipid bilayer. So any answers for this? Have you answered this question, any one of you? Yes? The reason why I'm asking is that these are so easy and direct questions that if you have a basic idea, you can definitely click onto this. Even if you don't know everything, you have one idea that gases will be diffusing freely across the lipid bilayer. With that one information, you can easily catch up the option 1. Now, there can be a reason, there can be a confusion between the diethyl urea and urea. What could be the answer? Another aspect is that ions, obviously, they cannot diffuse through the bilayer freely. They depend upon some kind of channels. So we can eliminate these options as well. Then lysine, amino acid, water, we have an aquaporin for that, glucose, we have glute transporters. Ethanol is diffusible, but compared to carbon dioxide, it is less. So the permeability of plasma membrane goes on like this. As you can see, gases will be diffusing faster. Followed to that, you will find ethanol is there, urea is there, water is there. Yes, Nagashri, absolutely correct. Then only you will come the impermeable molecules. That means they depend upon the transporters, ion channels, carrier proteins for the transportation. So the main difference which I was speaking about urea, as I told you, these are small, highly polar molecules because they can form hydrogen bonds with water. But on the other hand, if you will find diethyl urea, in their structure itself, you will find 2 ethyl groups are there, because of which it makes it less polar and more hydrophobic in nature. And we know that our lipid bilayer is made up of what? Hydrophobic fatty acid tails. So obviously they will favor the diethyl urea compared to that of the urea over here, right? Then this is regarding a unit 12 question. So every time we expect one question, but previous question papers and all, if you see, you got one or two questions, 2 to 3 questions also you got from the same topic. But this time it was very less, as expected, one. And that too, it came from transgenic plants. So here some statements are being provided over here. You have to identify the incorrect statement over here. So many a times we always lack this clarity whether they are asking correct or incorrect, because suddenly we see a question and we jump into an answer. So that should never be happening. Always read the question whether they are asking incorrect or correct statements. So in this case, you can see the first statement. Auxin can be used as a negative selection marker gene. Auxin is never used as a negative marker because it is simply a plant hormone. Marker genes are those which includes your herbicide resistance genes, antibiotic resistance gene, to check whether the cells have been transformed or not. So auxin will not come over here. Then moving on to the second statement, agrobacterium will be inserting the tDNA at random locations within the plant genome. That is true. See, they are not going for any complementarity. When the tDNA is coming from the bacteria, they just randomly integrated. We call it as an illegitimate recombination. Okay, so that is not dependent upon the homologous region. So this statement is correct over here. Next, what we are having, the antibiotic kanamycin. So they interfere with the cytoplasmic ribosomal protein synthesis machinery, thereby acting as a positive selection marker. They do act as a positive selection marker. But that is, again, if you say antibiotic kanamycin, they will be interfering with the prokaryotes. Even we have many experiments where it is shown that they interfere with the prokaryotic translation machinery. In this case, they are not doing so. So the statement D, gene transfer by biolistic or particle gun bombardment usually results in lower transgene copy number. No, it results in higher copy number compared to that of the agrobacterium mediated gene transformation. So you got the correct answers over here. I mean the incorrect statements. That is statement A, C and D. So very simple this one also. Even if you are not aware about A, C and D, at least you know the basic how the agrobacterium mediated gene transformation takes place. So with that you can click on to the answer. So that is what being mentioned over here. See, kanamycin used as a positive selection marker, but it interferes with the prokaryotic translation machinery. Similarly, the gene gun method, they result in higher transgene number and more of DNA rearrangement compared to that of agrobacterium. So that's why we have opted for statement A, C and D. Next. The next question is from unit 6, that is regarding the terpenoid biosynthesis pathway, secondary metabolites. So here they have provided with two pathways. One is the MEP pathway, another one is the mevalonate pathway, right? And they are provided with some statements over here, and you have to identify all the correct statement out of this. So what should be the correct statement over here? Any idea? In this case, the first statement stands true, that it occurs in plastids, which one? The MEP pathway, whereas the mevalonate pathway, they occurs in the cytoplasm. That is true. Now, if you see the statement B and C, right? If you know the basic idea that what does mevalonate pathway give rise to and what does MEP pathway give rise to, in that case, you can easily click on to the answer C. The correct answer over here is A and D. Why? C. The mevalonate pathway will be supplying the C5, that is carbon units, for sesquiterpenes and triterpenes. Okay. Similarly, the MEP pathway, they will be responsible for supplying their C5 units to both monoterpenes and diterpenes. So if these two concepts is clear, then yes, you can able to get into a 4 marks out of this question. Another match the following kind of question, which is most of the questions, if you see in this exam, you have come across match the following. And every time, whenever I take the session to students, I always say, never leave the match the following kind of question. Reason, you have some information about it. Just have a patience to read it thoroughly and remember it. Suppose, for example, over here. So here they have provided with some enzymes, and here they are provided with some hormones, plant hormones. So basically these kind of match the following every time you get in the exam. Sometimes they ask about receptors, sometimes they ask about precursors, sometimes they ask about their signaling pathway. This time they have asked about a specific enzyme that is relevant to the plant hormone. So what is the correct answer over here? The correct answer over here is fourth. See, if you see, when you look into these enzymes, first you have to identify which enzymes you are confident on. For example, I am pretty sure that among the 4 hormones, these 2 hormones will be familiar to all of you compared to other hormones, right? So first check it out how we can connect this. ACC oxidase. If you have studied the ethylene synthesis pathway, then you will be aware that ACC oxidase plays a very important role in the ethylene biosynthesis. Similarly, 9-cis epoxy carotenoid dioxygenase. Now, this is a very important enzyme that plays an important role in abscisic acid. Now, you got what is C and what is D? So, C is 4 and D is 3. So similarly, you got these option also. You can easily eliminate option 1 and option 3. This much you are confident. Among these two, if you remember that brassinosteroid receptor, where are they located? Then you can easily take up the ER localized cytochrome monooxygenase enzyme. So with that, you can even get the third one also, that is B21, which is not found over here. Now what is left? The last one, strigolactone. Now this you can connect it over here. That's it. So that's why I said, whenever you go for match the following, you'll find something that will help you out to get into an answer. So this is just an image where they have mentioned about the biosynthetic pathway of abscisic acid over here. Then this is the ethylene biosynthetic pathway where you'll find ACC oxidase, which is a key regulatory step when it comes to ethylene. That, like, it is a mandatory step. Without, if it reaches this ACC oxidase, then it cannot go further. Like, it will not go backward. It has to move ahead only. Moving on to our next question. So apoptosis. So every time I say apoptosis, even though it is a small topic under your subunit 4D cancer, but it holds a good weightage when it comes to the exam. Every time you get a 4 marks question over here. Now, what is this question? How many of you could attend this question, apoptosis? This was also a kind of easy question. If you have the information regarding the regulatory genes that are involved with apoptosis, how many of you could get into an answer for this question? Anyone? So here what they have mentioned, the human ortholog of C. elegans, that is SED9. So you all might be aware that we have got different SED genes and different vertebrate genes which function similarly, but their names are different from each other, right? So SED9 genes are overexpressed in a B cell lymphoma, right? So SED9 is considered to be a regulatory gene. If the regulatory gene itself is overexpressed, then you can imagine what will be the consequence of that. obviously it will lead to some kind of cancerous cell because it's a kind of gain of function mutation where the proteins will be produced in excess. Second point, if you see, SED9 gene, which undergoes gain of function mutation, or SED3 gene, which undergoes loss of function mutation. So in both the cases, both the mutation will lead to what? Cell survival or cell death? Tell me. Do you have an idea whether it leads to cell survival or cell death? Basically, if you think gain of function means obviously it will lead to cell survival. See, this is your regulatory gene. The regulatory gene will be holding the SET4 gene so that it will not be activating SET3 and it will be leading to cell survival. If the SED9 is activated, if the SED9 is being inhibited, it can be inhibited by any kind of pro-apoptotic protein. If this is inhibited, then your SET4 can activate the SET3 and that in turn leads to cell death. So if this undergoes gain of function mutation, if this too undergoes loss of function mutation, in both the cases what happens? It results in cell survival only. Right. So that's the reason you will find that there will be more than 947 non-gonadal cells will be there. Then on the other third statement, if purified EGL1 is added to SED9 SED4 complex, it will prevent the autocleavage of SED3, it is mentioned. How it will be preventing? It won't be preventing because EGL1 belongs to the BH3 only family of proteins. And you know that BH3 only family proteins are the one which serves as a pro-apoptotic signal. So if it is being added, instead of inactivation, it will be activated. Right? Now, the last statement is SED8, which is a multi-spanning plasma membrane protein. This is required for transferring the phosphatidylserine to the external surface. Yes, because phosphatidylserine moving to the external surface, that means to the outer leaflet, serves as a marker for a cell that has underwent apoptosis. So that's the reason A, B, D. These three statements are correct, whereas this is incorrect. Okay. So moving on to our next question. A researcher placed the plant cells in a hypertonic solution to cause the cells to shrink. Now they have provided some statements regarding the plasmolyzed cells. Now, what do you mean by a plasmolyzed cells? Any idea? What do you mean by a plasmolyzed cells? Any idea for that? Regarding that? Plasmolysis means what? When you call a cell has underwent plasmolysis, or how can you say that the cell has underwent plasmolysis? What is the condition for that? What is the condition? Plasmolysis is nothing but where the cells will undergo dehydration. Right? Excess of water will be lost, right? That is what we call it as a plasmolysis. So here some statements are given with respect to plasmolysis. Microtubules will be lost in the plasmolyzed cells. Protoplasts are retracted in the plasmolyzed cells. HETN strands are lost during plasmolysis. Patches of plasma membrane remains fixed to the cell wall. So what should be the correct combination of statements over here? Again, this is a part of unit 12, of course, but it doesn't require that you should study any specific topic to get into an answer. Simply, if you know what is a plasmolysis and the idea regarding the microtubules which you have studied in unit 12 and these strands, then it will be easy for you to get into an answer. So basically, it's a mix of both unit 2 and unit 5, we can say. So the correct answer over here will be the statements B and D are correct. So why the statements A and C are incorrect? Let's find out. See, microtubules, they are not completely lost at the time of plasmolysis. It simply undergoes the reorganization step. So that's why we call statement A as incorrect. 2 is correct because during the plasmolysis only the protoplast will be shrinked and it will be removed from the cell wall will be removed, because as I told you, plasmolysis means what? Dehydration, that is loss of water. So that is a correct statement. And this Histone strands, these are nothing but cytoplasmic strands connecting the plasma membrane to that of the cell wall. So at the time of plasmolysis, it do disappear, but immediately it will not disappear. It will remain. Only if undergoes a severe dehydration process, they will eventually break down. So that's why the statement C is incorrect. Then coming to statement D, yes, once your protoplast gets shrinked, there will be some patches of plasma membrane which will be remain attached to the cell. So that is basically mediated by some adhesive proteins or some adhesive interactions among the cells. Okay, so that's why the statement D is correct over here. I think you got a question regarding cadherin in Unit 5 also, right? If I'm correct. Unit 5, you got some question related to cadherin. Yes? Something related to hyposignaling, I guess. Of course, cadherin, again, it's a part of cellular communication, and hyposignaling something related to developmental biology, right? So here they have asked in terms of Unit 4C, that is cellular communication. What is the role of cadherin when it comes to cell junctions? So what are cadherin? You all might be aware that cadherin are calcium dependent adhesion molecules. So this is very important cell adhesion molecule because they are important during the embryonic development. That means when the embryo is growing in the initial stage, when it is in the fetal stage, when the new cells are being formed, new organs has been formed, at that time cadherin plays a very important role. So cadherin are calcium dependent adhesion molecules that contribute to cell interaction by forming homophilic interaction. That means what? This is one cell and this is another cell. So cadherin present in this cell will be interacting with the cadherin present in another cell. Right. So that is what we call it as a homophilic interaction. Similar kind of cells will be interacting. So that is true. Cadherins will function in tight junctions. No, it will not function in tight junctions. Can anyone tell me which proteins are involved in tight junctions? Which proteins are involved in tight junctions? Can anyone tell me? Which proteins are involved in tight junctions? Those are claudins, occludins, right? These are the proteins that are involved in tight junction. Of course, tight junctions are responsible for sealing the space between the adjacent cells, but they are not related to cadherins over here. Then N-cadherins are primarily found in the epithelial cells and mediate adhesion to the basal lamina. From the name itself, you can identify N-cadherin indicates neuronal cadherin. So how we can connect it with epithelial cells? If they have mentioned about E-cadherin, yes, we can connect. E stands for epithelial cells, but they have mentioned N-cadherin. Then cadherins are involved in the formation of focal adhesion linking the cell cytoskeleton to ECM. No. We have 4 interactions, right? One is focal adhesion and hemidesmosomes. Another set is adherence junction and desmosomes. So these are interacting through the integrin molecule and these two are interacting through cadherin molecule. So this is incorrect. Then cadherins can interact with actin cytoskeleton through associated proteins like catenins. That is correct because that is what happening here. Adherence junctions and desmosomes. So this can be a correct answer over here. So what we got? Yeah, statement A and statement E are correct. Right. Very simple, straightforward question if you have these much information with you. Next, we have the very important question. It may be confusing at times, but yes, this is an interesting question that came from insulin receptor, that is receptor enzymes. So many a times I say that nowadays the focus is given on receptor enzymes. Earlier it was like they used to ask too much of GPCR related questions, the mutations and all. But if you check out the previous year question paper, there also they have mentioned about FOXO related to a question. This time also a 4 marks question come. The insulin receptor is a receptor tyrosine kinase which engages with PI3 kinase pathway to regulate a FOXO transcription factor. So FOXO transcription factor, what is its regular function? Basically, they are involved with carrying out or regulating the process of gluconeogenesis or glycogenesis. Right. So basically, glycogenolysis. So basically, production of glucose. So a student uses QRT-PCR to determine the expression of FOXO target gene. Suppose they have taken a target gene, that is gene X, in a mammalian cell line under different conditions and makes the following observations. Treating the cells with P10 inhibitor increases the gene X expression. A cell line with an AKT mutation. What is that mutation? Serine has been changed to alanine at 308 position. This has increased the gene X expression. Changes in the gene X expression due to ligand binding defective insulin receptor is partly reversed by a P10 inhibitor. And lastly, phosphorylation of FOXO by PDK1 creates a phosphoserine binding site, thereby reducing the gene X expression. And here you have to identify the correct combination of statements. First thing to be noted over here is you have to be clear with what is a P10 is. Right. P10 in two ways we can describe. P10 is considered to be a tumor suppressor gene as well as a gene that is involved for termination of your PI3K pathway, PI3K AKT pathway, right? So what exactly it does? This P10 will be responsible for dephosphorylating PIP3 to PIP2, right? That means PIP3, one phosphate will be removed, PIP2 will be there. But this PIP2 will not be able to activate the AKT. Right, so the gene expression will be terminated. So in this case, what happens? If P10 is inhibited, obviously the AKT expression will be increased. So if AKT expression increases, there are chances that it will reduce the FOXO production. Right? And if FOXO production is reduced, obviously the gene X production is also reduced. Right? So that becomes your statement A. Now coming to the statement B, we have a cell line with an AKT mutation has increased gene expression level. So here the answers are statement B and C. Because the serine has been mutated and it is being replaced with alanine. And you know that phosphorylation will occur only in specific amino acid. So if that specific phosphorylation does not happen, then obviously it results in increased gene expression because there is nothing to control over there. Then the statement C, change in the gene X expression due to a ligand binding defective insulin receptor can be reversed by a P10 inhibitor. Yes, that can be done. And the statement D, phosphorylation of FOXO by PDK1. So, do you think so? PDK is the one which is responsible for phosphorylating FOXO1? No, right? It is your protein kinase B that is responsible for phosphorylating FOXO1. PDK1 is responsible for phosphorylating PKB. Right? So, this is phosphorylating protein kinase B. Protein kinase B is phosphorylating FOXO1. So, this is what the connectivity is. So, that's why you have to eliminate option D as well. So this is just the basic pathway of FOXO1 degradation. If you see, when the insulin goes and binds to the receptor, your PI3K kinase will be activated, and this PI3K kinase will be activating the protein kinase B, which will enter inside the nucleus and will phosphorylate the FOXO1 protein. So once it is phosphorylated, they will be exported out and targeted for proteasomal mediated ubiquitination. This happens when you have got insulin present, because when insulin is present, you don't have to increase the blood glucose. Insulin comes into play when we already have a blood glucose, right? So that's why it leads to the degradation, so that gluconeogenesis and glycogenolysis should be inhibited. So am I clear with this question, or is there any confusion? Am I clear with this question? See, if you have any doubt, whichever question you are not able to understand, you will get a link. In that link, you can have a check. You can post that question, and so that if you have any doubt on any question, you can let me know. Okay. Moving on to our next question. Now, this is regarding the intracellular protein transport mechanism. So you have different receptors kind of things mentioned over here. Here also you have to identify the correct combination of statements. So the proteins that are destined for lysosome are tagged with the mannose-6-phosphate group in the Golgi apparatus. Why? Because the corresponding receptor is located in the trans-Golgi network. That is correct. Next, what we are having, the signal recognition particle. They will mediate the insertion of proteins into the mitochondrial membrane. Is it correct? Does SRP is responsible for inserting the proteins into the mitochondrial membrane? Is the statement is correct? No, because SRP is related to your endoplasmic reticulum. Whereas for mitochondria, you have got that TIM-TOM complex that is responsible for the translocation, right? But in this case, it is only the SRP molecule. Then what you are having, the Kedal receptor. Kedal receptor in the ER and Golgi works by retrieving the soluble proteins accidentally which has moved to Golgi. That is true. The Kedal receptor which is present, they shuttle between these two compartments. Why? Because if by mistakenly the protein moves ahead to the Golgi apparatus, which is supposed to be stayed within the ER only, in that case, it can be retrieved back. So this statement is correct. Next, what we are having, the cargo proteins that needs to be exported from this ER, cargo proteins that needs to be exported from the ER are packaged into COP2 vesicles based upon the presence of ER export signal in their cytosolic tail. So you might be aware that we have different COP vesicles involved, COP1, COP2 and clathrin. COP2 will always move in forward direction. That means they move from ER to Golgi because their origin is from ER. And COP1, if you see, they move in the reverse direction from Golgi to ER. Clathrin is a different one. So this statement is also correct. Then moving on to the statement E, the clathrin-coated vesicles are primarily involved in vesicle trafficking between Golgi and ER. No, they are not involved with Golgi and ER. Do you know it is involved with which two cell organelles? Anyone remember? Clathrin-coated vesicles is involved with which two cell organelles? Anyone remember? Sure, Ankita, you can post this question ID in the link which will be shared with you shortly. Or if it is already being shared, you can post it so that I will get into an answer. Yes. Anyone knows clathrin-coated vesicles? It comes under which two cell organelles? See, it basically travels between Golgi to endosome, plasma membrane to endosome, like that. So basically, it's a connectivity between trans-Golgi and endosome vesicles, or it will be between a plasma membrane and endosome vesicles. Okay. So this is a table which, if you remember, it will be easy for you to understand. Yes, Nagashri. Moving on to another easy question taken from solute transport, that is a kind of match the following. I am pretty sure that you should have you have given the correct answer for this, if I'm correct. Have you all? Just can I get some answer for this question? Yes, if anyone can give me the answer for this question. Very easy question. ATP powered pumps, uniporter, channels, symporter. And on the other hand, their properties, or I can say the definitions, are given. So how can you get into that? Okay, I will check on to that question, Ankita, for sure. Yes, what should be the correct match over here? I think someone should have answered this question. This is such an easy question. So the correct answer over here is very simple. It's your option 4. So how it is your option 4? See, ATP powered pumps, as the name indicates, what it will do. It will take the energy and then it will move against the concentration gradient. And uniporter means what? Basically, yes, Nagashri, and another one, 0.6. Okay, yes. Kumar, just have a look over here. Uniporter means, yes, they will be moving in a single direction. So they will be transporting the hydrophilic molecules down their concentration gradient. Single orientation it will be. Next, you have got the C2, that is channels. Channels, it will behave like it is an ion channel, so obviously free diffusion of molecules will be taking place. It is not like down their concentration gradient or against. It is just a channel that allows the movement of ions. And the last one is the symporter. Symporter, if you remember the SGLT one, sodium glucose co-transporter mechanism. So what happens over there? The movement of molecules will be taking against the concentration gradient, which will be coupled to another molecule which is moving down the concentration gradient. The SGLT1, sodium glucose transporter, is the best example for that. Okay. So this is just a comparison of different transport mechanisms. If you can see, channels means they can be closed, they can be open with some specific stimulation. You have 3 types of transporters. Uniporter, if single molecule is moving. Symporter means 2 molecules are moving in the same direction. Antiporter means 2 molecules are moving but in different opposite direction. And ATP powered pumps, if you see, they will move against the concentration gradient. Okay. Next, another direct question based on match the following. Now, this is regarding the cytoskeletal system. Has anyone answered this question? Because nowadays, again, the trend regarding the cytoskeletal system has changed to the accessory proteins, which I regularly, whenever I take any session on the CSIR NET, I always say, focus on this particular accessory proteins. Nowadays, it's a trend and you always get a 4 marks out of it. Last time also you got, this time also you got. Any answer for this? Do you have any idea for this question? Anyone has attended this question? What is formine? So this is, they have completely asked about actin accessory proteins. Sometimes they will mix up actin, your microfilament and microtubule mixed up questions. This time they have only asked the actin dynamics. So what should be the correct answer over here? Anyone has given the answer for this question? Formine, ARP, 2/3, coffeeline, tropomyosin. What should be the correct answer? So here the correct answer will be option 2. So formine is responsible for nucleation only, and they will remain attached with the plus end. Whereas ARP do 3, they are also responsible for nucleation, but the difference is that they will be going for branched network. That means they will keep on forming the branches. Coffeeline is responsible for breakdown, or you can say like disassembly. That's why they go and bind to the ADP actin filaments, accelerating disassembly. And the last one is tropomyosin. Tropomyosin, as the name itself you can understand, they are responsible for stabilizing the overall filament, right? So this becomes the answer for the question. For to crack these questions, you have to be very much clear with the accessory proteins. Formin and ARP2 bar 3. The only difference is that here they will form branches. Then you have got thymosin. Then you have got profilin that helps in elongation. Thymosin, they will undergo disassembly. Tropomodulin, disassembly. Myosin stabilizes. Cophylline, ADP actin filament. So when I say ADP actin, you can clearly understand that they are saying about disassembly. Capping, they also prevent assembly and disassembly. Gelsolin, they will break down. And these are some other accessory proteins that can help in bundling, cross-linking, or attachment. Then another good question that came from membrane structure and function. So an experiment was carried out. Now this is like, if you know the difference between your peripheral membrane protein and the integral membrane protein, if you know the difference, you will be able to get into the answer. Protein X can be extracted from disrupted erythrocyte membrane with higher salt concentration. Treatment of intact erythrocytes with protease followed by their extraction led to the intact protein X. Treatment of disrupted erythrocyte plasma membrane with proteases followed by their extraction led to the fragmented protein X. So these were the interpretations done. You have to identify the correct interpretation among that. So what they are saying, they are able to extract the protein by a simple protease treatment. So that is giving you a clue to get into an answer. What is the correct answer over here? The correct answer is A and D. You all might be aware that peripheral membrane proteins are the one which is just bound to the membrane. So any basic treatment you give, any pH change or like this, the protease treatment, something like that, then what happens is that they can be extracted easily, right? Integral membrane protein, they are attached to the they are attached via hydrophobic interaction. So you have to provide with some detergents to extract it. Protein X is present on the extracellular matrix of the membrane. Protein X can be present in the cytosolic phase of the plasma membrane. See, here they have mentioned treatment of intact erythrocytes with protease followed by extraction led to the intact protein X. So they have done some work. Treatment of disrupted erythrocyte plasma membrane with protease followed by extraction led to a fragmented protein X. So based on these statements itself, you can get into a conclusion over here, whether it is a peripheral or integral, or it is an extracellular or cytosolic. Sometimes the answers are there in the question itself. The only thing is that you have to read the question thoroughly to get into an answer. That is the plan over here. That is the easy technique over here. So that's we have done with the morning shift questions. Moving on to the evening shift questions. So how was the morning shift questions? I hope so you were able to understand. If you had made any mistake, what can be an approach to get into a correct answer? I hope so this is getting this, you are getting a huge help out of this, if I'm correct, right? So moving on to the evening shift, again the very first question. Can anyone tell me, such an easy question, among the phytohormones, which of them will have the two component system? Which one? Very simple question that has come in evening shift paper. What should be the answer for this question? Phytohormone signaling pathway. Which hormone follows the bacterial two component system. I hope so you are aware about bacterial two component system. That is, they will have an histidine kinase activity, plus they will have a response regulator. So this will undergo autophosphorylation, transferring the phosphate over here. Usually here aspartate residue will be sitting. Here histidine is there, so this will be transferred over there. So that same thing is applied over here also for cytokinin and ethylene. These are the only two plant hormones which focuses on bacterial two component system. Okay. Then next question is regarding cancer, that is haploinsufficiency in tumor suppressor genes can be caused by all of the following mechanisms except deletion of one allele of a gene, a missense mutation that leads to increased expression of one allele, nonsense mutation in one allele leading to truncated protein, or epigenetic silencing of one allele. For attending this question, you have to be clear about what is an haploinsufficiency is all about and how can we connect it with the tumor suppressor gene. That is first point over here. Second is be clear with the terms. Of course, deletion anyone can understand. What is a missense mutation? Where a single amino acid is being replaced. Nonsense mutation, which terminates the protein prematurely. Epigenetic silencing, that is the changes that happens in the DNA and histone levels for regulating the gene expression, right? That is epigenetic changes. Now you have to connect this with haploinsufficiency. The correct answer over here is a missense mutation leading to increased expression of one allele. So what is that? Let's see. First thing is that haploinsufficiency means what? When a single functional copy of a gene is insufficient to maintain their normal function. So it is at that stage it causes disease. So in case of tumor suppressor gene, haploinsufficiency promotes cancer development. So deletion of one allele will reduce the gene dosage by removing one functional copy. Nonsense mutation, this is one allele leading to truncated protein results in non-functional protein from the mutated allele, thereby reducing the functional gene product. And last one is the epigenetic changes, which causes silencing of the gene. So what we are left is missense mutation. So this is the only mutation which increases the gene expression, leading to haploinsufficiency. Okay, so it not only reduces the functional gene dosage, rather it increases the protein function activity. Missense means I told you a different amino acid is being replaced. So in this case, the protein activity will be altered, increased production will be there. So it involves a reduction in gene dosage, not an increase. Okay. Now moving on to a very simple question. This is regarding the protein glycosylation. And in this part, you can see that very direct question they have asked. Right? I guess you might be able to answer this question. Right? Very simple question. See, the correct answer, if you remember the different sugars over here, it will be easy for you to remember. See, this is your N-linked glycosylation, right? So you have got 2 NAG attached, 5 mannose in cytosol, 4 mannose in lumen. So total 9 mannose, then 3 glucose. So you got an answer. You don't have to remember every step, just remember the number of sugars. Then which of the following describes the function of snare proteins? Snare proteins are always used for fusion of a vesicle to that of a target, because we have the corresponding V snare present in the target and a snare present in the in the vesicle is present, target is present. So they will undergo fusion, as you can see, with some accessory proteins, and the membrane fusion takes place, the content will be released. So that is what your membrane fusion is all about. Then this is a 4 marks question which is related to your plant. As you can see, these are 3 conditions given. the light direction and the movement of chloroplast. This is the same image given in the Tays and Ziegler book also. Very direct question. So they are asking you which of the following combination correctly matches the chloroplast distribution with its corresponding light intensity. So here you can see the chloroplast is accumulated above. Here you can see they have gone downward, and here you can see they are tethered to sidewards. So how we can interpret this? See, the correct answer is A is low light. So that's why they're trying to grasp maximum of light. B is darkness. So too much during the darkness, they don't have any specific function. So they settle down. And C is the highlight, because if too much light is given to the chlorophyll, the cell organelles will be damaged. So they will move in the sideward. So during low light intensity, chloroplast will accumulate at upper region. We call it as a chloroplast accumulation response carried out by FOT1. Similarly, at high light intensity, chloroplast will go to side. We call it as a chloroplast avoidance response carried out by FOT2. See, this was the image I was speaking of. Exactly the same image is copy pasted in the question paper. Right? Very simple and easy. Moving on to our next question. Again, this is regarding the auxine transport in plants. So it proceeds via symplast. Its velocity is faster. Does it proceeds via symplast? Do you think so? Yes, Nagashri, you are correct. No, polar auxine transfer always passes through apoplast. The transportation takes place, that is through the cell wall and all. Its velocity is faster than phloem translocation rate. No, it can be faster than diffusion, but not faster than phloem translocation rate. Then it is specific for active auxins, definitely. Both natural and synthetic auxins can be moved across the mechanism. Then it is carried out by some carrier proteins. If you have studied the auxin transport mechanism, you might be aware about influx proteins like your OX, II, IAA, then the efflux proteins, which is present in the bottom of the cell, that is plasma membrane. We call it as a pin proteins. So these are the carrier proteins that helps in the movement. So that's why statement C and D is correct, others are incorrect. Next, again, a question came from apoptosis regarding a small experiment was carried out over here. So what happens is, while screening for loss of function mutants, a student found a mutation in the gene encoding caspase-9 in intrinsic pathway of apoptosis. These were the consequences that we are seeing. You have to identify what are the correct consequences that is going to happen if caspase-9 is mutated, right? So first thing is that the correct answer is B and D. So why the statement A and C is incorrect? First thing is that loss of mitochondrial membrane potential and release of cytochrome C has no connectivity with caspase-9 because it happens before the activation of caspase-9. So this cannot be possible. Second is inability to activate the death receptor. So this is related to your extrinsic pathway, not the intrinsic pathway. Right. And apoptosome formation is correct. Then become resistant to UV irradiation. Yes, that is also correct. Because see, if you compare, these are the radiations, toxins, and hypoxia. These are considered to be the initiating factors to carry out the apoptosis. Like that extrinsic pathway, you have got the death ligands and death receptors. Perforin, it is specific for the CTLs, that is cytotoxic T cells. Okay. Next question is regarding cancer. So here some statements are given to you regarding the role of tumor microenvironment contributing to the metastatic process. Here also you have to identify the correct combination of statements. So what is the correct answer over here? It is your A and C. So hypoxia in primary tumors can induce the expression of VEGF. VEGF means what? That is responsible for angiogenesis, that is blood vessel formation. So basically the hypoxia condition will activate a transcription factor known as HIF, that is hypoxia inducing factor, which in turn activates the VEGF and promotes the metastasis. Statement B, tumor-associated macrophages always inhibit metastasis. No, it can promote as well as it can inhibit. That depends upon the type of macrophages. Then, cancer-associated fibroblasts provide structural support and secrete factors that promote metastasis. Yes, they will be secreting the growth factors for carrying out metastasis. Then the last statement is acidic pH of the tumor environment impedes the cancer cell migration. It is not impeding, rather it will help in cancer cell migration. So that becomes the correct answer over here. So A and C are correct. Then a very simple question of match the following. If anyone can answer this question, very simple question. So here you have to identify which are correctly matched with the amino acid residues they can modify or they can act on. What should be the correct combination over here? Undoubtedly, it has to be A and B only. So, phosphorylation, as I told you, in prokaryotes, it happens on histidine. So, that becomes the correct. Ubiquitination, they happen in lysine. In some cases, yeah, bacterial cases, N-terminal methionine can be taken. O-linked glycosylation occurs in serine and threonine. Hydroxylation can occur on proline, but it doesn't occur on cysteine. It can occur in lysine as well. So that's why this is the incorrect one. A and B is the correct one. Okay. Then this was an experimental question. Adding mRNA that encodes a eukaryotic secretory protein to a cell-free translation system initiates protein translation. So signal recognition particle in low concentration and ER treated with 1% Triton X100, we are sequentially added to the cell-free translation system. What will be the outcome of that? So two things you have to focus on. One is SRP in low concentration, one treating the ER with Triton X100. So the consequence will be protein synthesis will start but terminate prematurely, leading to shorter products. The reason is that, see, we know that SRP plays an important role in recognizing the signal sequences, right? So if the SRP levels are low, obviously they cannot bind to the signaling sequence. So ribosomal targeting will be impaired. Now you are adding the Triton X100. So this is something that disrupts the membrane integrity. So the ER will be disintegrated. So whatever the protein will be generated, either it will be a non-functional protein. So combining these two factors together, we can say that ribosomes will stall and it results in premature termination, thereby producing the truncated protein. So here also the answer is there in the question itself. Then this is regarding the clathrin-mediated endocytosis. Very easy question again, vesicular transport. So some statements are given. You have to identify the correct combination over here. Clathrin-mediated endocytosis, I told you, it helps in the recruitment of adapters so that it can connect to the cytoplasmic phase of the membrane. Then low pH environment is mandatory because they can separate the cargo and the receptor. Multivesicular body pathway is also correct. What is incorrect is this one. Late endosomes which matures into the lysosomes are directly involved in the recycling of synaptic vesicles. Endosomes are not involved in recycling. They are involved in the cell death pathway, right? So recycling of synaptic vesicles, they are not being carried out. In fact, recycling of synaptic vesicles, you have separate set of proteins, accessory proteins related to SNARE that helps in doing so. Okay. So this is just an image for LDL particle, how the receptor and ligand is getting separated inside the endosomes. Then this is regarding the cholera toxin. Very easy question. Another easy question over here. So they are saying that cholera toxin activates the chloride channel in the epithelial cells of intestine, which leads to the chloride ions and consequent loss of water, leading to dehydration. So for that, ORT is given, that is oral rehydration therapy is given. So what is the purpose of this ORT? Obviously, it will create an osmotic gradient with sodium and glucose that will allow water to move through the intestinal lumen via epithelial cells. Now, here also, which transporter will be acting on? SGLT1. Okay. So this plays a very important role over here. And this is the last question. the lipid composition of the two monolayers of plasma membrane is quite different, and the lipid asymmetry is functionally relevant, especially in converting extracellular signals into intracellular ones. So here few membrane lipids are given: phosphatidylserine, phosphatidylinositol 4-phosphate, phosphatidylcholine, and sphingomyelin. Now you have to identify that correctly defines all the lipids which are involved in signaling and are also restricted to the cytosolic phase of the plasma membrane. That means a lipid which is present in the inner leaflet of the plasma membrane at the same time participate in signaling also. What should be the correct answer over here? It should be A and B. So serine is located in the inner leaflet of the plasma membrane, and this plays a very important role in cell signaling process, specifically the PI3K pathway. Even in GQ pathway, you see the phospholipids are playing the role. Then phosphatidylcholine, these are present in the outer leaflet. Same is the case with sphingomyelin, and they don't have any specific role when it comes to signaling. Okay. So here we have come to an end of the session, and I hope so this session was really helpful for you to identify the pitfalls. That means where you have gone right and where you have gone wrong. If you have went wrong, no problem, you can always work on towards it and get it yourself into a better thing next time whenever you attempt this question. So in the next week you'll come across rest of the units with other faculties. You will come to know about other units as well. Okay then, thank you so much everyone. Good night and take care.